Can we go? Okay, so welcome to the second day of this conference. My name is Manuel. I'm sharing this morning session together with Aris.
Dermitzakis.
Yes, so we are looking to three exciting talks, and finally then, before the coffee break, we have an announcement, and probably we will put 5 minutes from the coffee break in addition. So the first talk is about giant hydride polymer lipid vesicles, phase separation and dynamics from advanced fluorescence and microscopy methodologies, and the speaker is Manuel Prieto. Yeah, please, Florence.
Thank you so much, and good morning, everybody. And it's a great pleasure just to be here, and I would like to thank the organizers for the kind invitation. Also, this was on behalf of the IFMBE International Union of Current Applied Biophysics, of which Anthony Watts, which will follow me, the president, will address. So, I'm sorry, my voice is still not warmed up. It will improve. But I will talk about giant hybrid polymer vesicles and addressing essentially phase separation and dynamics as taken from advanced fluorescence and microscopy. To put in context, if we pick up a polymer, and this polymer has in the center some hydrophobic section and two hydrophilic moieties outside, these two polymers, they can assemble in these entities called polymer zones. And these polymer zones, they have high stability, advanced functionalization. However, a limited biomimetic character. On the other hand, we have other entities which are called the liposomes. So phospholipids, they self-assemble, making the double layer. And these liposomes, they have been certainly the model for the biological membrane, and most of my life has been carried on them. And they have a very high, certainly, biomimetic character, although no mechanical stability. Liposomes, they don't stand the high shear stress when going into very thin blood vessels and all that. So the idea is just to make some kind of hybrid vesicles. This is picking up amphiphilic copolymers and phospholipids and self-assemble them into these entities. The idea is just to get, how can I say, the best of two worlds regarding properties. And in these entities, we all have this red area, which is lipid, which is very compatible. We all have anasama in red. In green, we have copolymers, which is very robust. And into the lipid part section, membrane proteins, they can be inserted. Anyway, we have these two situations. We can have either an homogeneous membrane, I mean lipids, they are homogeneously distributed to be these red spots into the polymer part, and certainly that you can have phase separation, and I will come into detail to this equation. We have here polymer and lipid in the very center. So these entities, they have been used in molecular recognition, direct typing, etc. I'm not telling you about applications, but I will present some detailed physical characterization of these entities. So for these entities, both giant vesicles, which can be seen under the confocal microscope, and large hybrid vesicles, they will be used, considering how they vary according to the polymer molar mass and architecture. And also properties, I will mention, for instance, fluidity in these systems, related to diffusion, as we all know. There are two very important considerations that I will take into account. And one is the hydrophobic mismatch. Here we have the polymer and the lipid in the center. And at this interface there is a line tension, which is an energetic penalty. By one reason, Here we can have, for instance, water in contact with the hydrophobic part of the polymer. Another consideration that will come later is the so-called bending energy. We can bend a membrane, okay, but in that case we should spend the bending energy which scales with the square of the thickness of the membrane. So let's see the type of polymers that we have used. Triblock polymers that were used, they have this central section which is hydrophobic, made of polydimethyl siloxylane, and the two hydrophilic extremities from polyethylene oxide. There it is, the membrane. The three polymers, they were fully characterized regarding several techniques. For instance, the hydrophobic thickness was here evaluated from cryo-TM, and from now on I will mention the shorter polymer, the intermediate, and the larger polymer. Here they are, the thicknesses of the membranes, and also aggregation numbers and also areas they were determined according to light scattering and other methodologies. In addition to these triblock polymers, another polymer will be mentioned, which is a graft polymer. And this graft polymer, it has a different architecture, and this will be compared. So here they are, the three polymers, shorter, intermediate, and large one. Here it is, the graft polymer. The graft polymer, it can also make some kinds, it can make an hydrophobic interface here according to some hairpin configuration. And these polymers, they will mix up with lipids. One lipid is PLPC, a phospholipid component, by far the most important fluid lipid, and also DPPC as a model for the gel phase lipid. The phase transition is higher. So let's see how it is the phase separation of lipid and polymer phases. These giant vesicles, they were made by the standard electroformation. It's very easy and it allows, let's say, a very quick way of obtaining the giant vesicles. And to visualize them under the confocal microscope, the polymer was derivatized with fluorescein, which has a green fluorescence. So here you can see this is a poor polymer zone, and we have an homogeneous green fluorescence. For visualizing the lipid part, this lipid OP derivatized with rhodamine is used. And on the right we have this red fluorescence from the lipid homogeneous. Now, if we could dim the lights a bit, perhaps it could be nicer for the figures. Now, when making hybrid vesicles, we have the two following limiting situations. We can have an homogeneous distribution of lipid inside the polymer. Here it is, we have this, it's homogeneous, and we have this yellowish color, which is the result of green and red, or we can have phase separation. Please see on the right we have here clearly green part which is made of polymer, and on the right we have the red lipid part. So in this case there is phase separation of the two entities. So let's see how it goes for the first shorter polymer. And we'll appreciate the types, the entities that we obtain upon increasing the lipid concentration here on the bottom according to temperature. These are not real phase diagrams, since the system is not strictly under hydrophobic equilibrium. And for the shorter polymer, for the short lipid concentration, we have homogeneous distribution. These are the equatorial projections under the microscope, and well, they are yellowish. We have homogeneous distribution of lipid inside the polymer. Upon increasing the concentration, we have stable domains. Well, here we have beautiful phase separation. You can see the green polymer and the red lipid in both cases. Keeping on increasing the concentration, we have this phenomenon of budding. So budding can be either from the lipid, as shown here in red, or the polymer here in green. Why budding? Budding, it happens, I'm I mentioned before that at the interface we have line tension. And so, if there is a competition between line tension and the bending rigidity, see this case. In case that we have a very long domain edge, and if we create a bending, the domain edge is going to decrease, so line tension decreases. Certainly we should spend the bending energy that I mentioned before, but the system minimizes the energy upon bending, and this is the reason for the bending processes. And due to the very high liquid concentrations, well, there is domain fission and the system becomes unstable. Now, intermediate size polymer. Intermediate size polymer, here the mismatch between lipid and polymer is larger, as we see here from the different dimensions. And it's very interesting, since we have the so-called homogeneous distribution up to a much higher concentration of lipid. We can understand it. Since the line tension is greater due to the greater mismatch, the system tries to avoid phase separation, which would be an energetic penalty. And well, phase and budding, it only comes up at higher concentrations of lipid, as we see here, as compared to the first polymer. And again, to very high concentrations, it's domain fission. Now, let's move to the very low polymer as compared to the membrane. Here, the hydrophobic mismatch is too high, and when we mix them, essentially, no hybrid vesicles they are formed. As you see here, we have poor polymer zones in green and poor liposomes in red, and just the small entities, they are mixed up. Line tension in this case is too large. Related to that graft polymer that I mentioned before that makes this hairpin configuration, although its thickness is not very different from the so-called shorter tribord polymer, in fact it is more homogeneous as compared to it. So altogether we have this equation. From left to right we are increasing the concentration of lipid, and from the bottom to the top, this is the sizes of the polymers. So shorter polymer, here it is, homogeneous distribution and stable microdomains. For the intermediate polymer, the homogeneous distribution can go up to higher concentrations to avoid the line tension, as I mentioned before, and for the top one, essentially we have separate entities and not statistically a very large number of domains they are, of hybrid vesicles they are obtained. And the graft is more unstable as compared to the tribord. So well, you cannot see it, but here in these lower lipid concentrations, we say that they are homogeneous distribution. But please note, there is here an interrogation mark. It's not very visible here with the light. And the interrogation mark is the following. Okay, under the confocal microscope, this looks like homogeneous, so we have no microdomains. But do we have nanodomains that we cannot see under the microscope? And so let's look if we have nanodomains. So in this case, this would be some pseudo-homogeneous distribution. For looking for nanodomains, let's go to FRET, Foster resonance energy transfer, carried out under the microscope, FLIP, lifetime imaging, and FRET basically is the following. We have one molecule, so-called the donor, which is excited, and it can transfer the energy to a second molecule called the acceptor. If they are, let's say, at short distance. In fact, the efficiency of the process is a dipolar interaction, and it scales with the inverse 6th power of the distance. So, okay, we have FRET if they are near. If they are too far apart, there is no FRET. Let's look into this cartoon what we can expect into the hybrid vesicles. In case of homogeneous concentration, here they are the red spots of the acceptor distributed within the lipid. They will be in close contact, and we have a very high efficiency of energy transfer. So the lifetime of the donor is decreased in the presence of the acceptor. In case that we have phase separation, as shown here, they are too far apart, and the lifetime of the donor it will be essentially insensitive. So let's move to the data. First, on the left, let's study a poor polymer zone with no lipid, just a very small amount of the acceptor. And this is the lifetime of histogram for the donor, the histogram of for the lifetime of the donor molecule. Well, it's about 3 nanoseconds, and in the presence of the acceptor there is a very large decrease. So we have, in this case, a very efficient energy transfer, so acceptors, they are close to the donor, such as I mentioned. Now, let's study a hybrid vesicle, and this hybrid vesicle is for the lowest lipid concentration, 10% of POPC. And in this case, as you can see here, there is a minor variation in the lifetime of the donor. So, this is clear evidence for the formation of nanodomains under the pseudo-homogeneous phase that we see under the confocal microscope, which resolution is not better, let's say 250 nanometers at the best. So this is the variation of the efficiency for the shorter polymer. After, let's say, 10% of lipid, clearly nanodomains, they exist. The effect is not so pronounced for the intermediate polymers, so less domains they are formed. This also can be seen by a micropipette aspiration under the microscope, and it's very nice. Sorry again, it's not very clear for you, all to the ambient light. And here it is. Here we have a hybrid vesicle under low suction pressure. And the lipid was labeled again with a lipid probe. So we have this red fluorescence. Upon increasing the membrane pressure, we see that clearly there is strong formation of this yellowish, of these red spots. So, increasing the suction pressure, we are inducing the coalescence of nanodomains into visible microdomains. And this is again to reduce the line tension. Very interesting result. It's a cartoon that came in the literature and is the following. And the authors, they pick up a system where they have coexistence of phases under isotonic conditions, and they put it into hypotonic conditions. Okay, this case is unstable. Water goes inside the vesicle and the size increases, so pressure also increases, and this is going, such as here in our experiment, this is going to induce phase separation. The domains, they start coalescing, just to decrease the domain edge. In the very end, we can have water leakage, and this comes back to the beginning. And this can cycle a few times. It's really very interesting. So, nanodomains, they coalesce under pressure, just to avoid it. Graph polymer, altogether, this is the summary. So, upon decreasing the molar mass of the triblock polymer, we have increasing possibility of having stable nanodomains. Graph polymer, no stable domains. And this is microdomains that I showed you under the microscope. And also very important, nanodomains, they are present and they are metastable and they are coalescing. Now, this was everything I told you. It's giant vesicles, they are very large, micron size, they can be seen under the microscope. Now some data about large unilamellar vesicles. Large unilamellar vesicles, they were here made using two different methodologies. In both cases, the final important step is that they are extruded through a 100 nanometer filter. So we get this we got these entities which are very homogeneous and much smaller, large unilamellar vesicles, and they cannot be seen under the microscope, as you can imagine. Let's study them. In this case, the question is again, do we have nanodomain separation in the large unilamellar vesicles? We come to FRET again in this case, and a very detailed FRET parameterization was done. Here you see that the polymer is thicker than the membrane, so we have to consider the donor loss that exists for called MDD, and also there is an interplanar distance and radius of exclusion. All this was taken into account in FRET modeling, and this is the equation taking into account FRET energy transfer from the two wavelengths. Here it is, the data. So again, I'm sorry, we have here two shaded areas. One is here in the top, and the second shaded area is here in the bottom. What I show here, these are the theoretical expectations that we have for FRET according to the formalisms of energy transfer, and these two limiting situations are the following. In the wide uncertainty of the shaded areas, this is due to the uncertainty that we have in the polymer area. So they were described like this, in this way. And we have two regions. One, it would be the theoretical expectation for homogeneous distribution. This means no nanodomains they are existing, or otherwise they would be too small to be detected by FRET methodology. This means in the range of 5 nanometers. Here, the second theoretical expectation is the one considering that we have nanodomains. In FRET language, we call it the infinite phase separation. And as you see, our data was according to this second provision, in all cases, just for deriving here the acceptance density, for deriving the correct formalism, the acceptance density was determined correctly. I mean, for the homogeneous distribution, total area of the surface was considered, and for the infinite phase separation, we should consider the partition coefficient of the lipids within the polymer phase. So the answer is yes, we also have nanodomains in large unilamellar vesicles. This is cryo-TEM data showing these entities, and please appreciate here. We can see we have here a poor polymer zone. This is the thick polymer membrane. This is an hybrid vesicle. Please see here on the right the thicker polymer membrane, and here on the left the thinner lipid membrane, and another hybrid vesicle over there, thicker in that upper section and thinner lipid section in the bottom. So in large unilamellar vesicles, we have also more efficient mixing according to the decrease of line tension, such as we do. Now let's consider a physical property of the hybrid vesicles, and we studied fluidity, which is certainly related to diffusion. This was carried out under the microscope in the giant vesicles and using some technique called fluorescence recovery after photobleaching. And it's very easy, it's the following. At time zero, we have a low intense laser pulse, and we select here some region of interest under the microscope, and we have fluorescence of the molecules. And here it is, the signal from fluorescence. At time zero, we give a very short, intense laser pulse that is going to photobleach to destroy the molecules, let's call. And so fluorescence intensity decreases to 0. After, we have free diffusion from the molecules. Sorry again, the pictures, they are not very visible under the light in the room. So after, what happens is the following. We have free diffusion of fresh molecules, fresh fluorescent molecules that come to that region of interest. And so we have a recovery of fluorescence intensity that goes with time. From the recovery of fluorescence intensity, I'm not showing here the equations, we obtain the translational diffusion to the students in the audience. We feed equations to the data and not the other way around. And from the diffusion coefficients, we can also obtain viscosity in the framework of Safman-Goldberg theory. So this is for diffusion of a cylindrical entity in a plane, that is the viscosity of the medium at the prime outside the medium. So let's see the diffusion of the three polymers that I told you, the short, intermediate and large one. And no big surprise, upon increasing the size of the polymer, the diffusion coefficient is decreasing. As expected, they are larger and larger and larger. Thank you so much. Now it happens that if you look now to the hybrid vesicles, let's see the diffusion of the polymer. And this is very interesting. The diffusion of the polymer starts decreasing, as we see here, upon increasing the lipid concentration. I showed before that here, for a small amount of lipid, we have nanodomains, and after we have microdomains. So, what happens? The lipid is diffusing, but we have nanodomains. So, let's say the lipid collides with the nanodomain, and this is going to delay its diffusion. So, this is the effect of the nanodomains upon the diffusion of the lipid, of the polymer, polymer colliding with lipid nanodomains, the data again over there. Now, this is less relevant as we could infer for the intermediate polymer. Nanodomains, they are less present up there. And now, this is very interesting. If you look to diffusion of the diffusion of the lipid is not affected at all by the presence of lipid domains. So there will be some fast exchange of the lipid when he encounters the nanodomain, and diffusion goes on. So it's practically insensitive. So the dynamics of molecules is dependent on composition and morphology, and diffusion is affected by the presence of lipid domains. Briefly, I showed you data obtained under the microscope both for giant vesicles and fleet, and for quadrilamellar vesicles also thread. And detailed characterization was made. Detailed characterization was made according to the size of the polymer's architecture, and we have truly verified the hybrid character of these entities. So they are ready for applications, and all in the literature they have been used. I came to the acknowledgements when I was studying photophysics lessons for the first time. I picked up this book, Photophysics of Aromatic Molecules by John Burks, in fact from England. And he made the acknowledgements in this beautiful way. He was saying that the work was carried out with a little help from our friends. This is a song by the Neanderthal band that the students in the audience never heard about, called the Beatles, and also the work my friends they are pictured within. Please, the students, go and see who is about the Enigma variations by Sir Edward Elgar, not a pop star. And here it is. This was carried out by fantastic a student from Vietnam. She was incredible. Thuy Tao. She made the PhD thesis in between our lab in Lisbon and another lab in France, in Bordeaux, in the framework of an European grant. The publications are described in the abstract book. So that was soft matter, ACS macro letters, and language, about 5 publications. And another collaborator is Fabio Fernandes, a senior member from the group in Portugal. And thank you for your attention.
Thank you very much for this nice talk. And I think you handled it good with the light in order to explain what should be visible.
Aris, yes, not very visible, the domains really.
That was okay. Yeah, we have maybe time for a quick question. Yeah.
Can you hear me?
Yes, yes.
Yeah. The question is about the applications, because I saw you had a slide about how these polymer vesicles can be used. Could you please tell more about that?
Yes, As I mentioned, we carried out no study of the applications. This is a study of the physical properties, or physical characterization and everything. But they have been used there are a significant number of papers in the literature studying its application, certainly in drug delivery, trying to have a specific targeting upon functionalization and all that. But it was not object of our study. How far this goes into applications, the hybrid particles, really, I'm not an expert. And for instance, liposomes, the poor liposomes of lipid, there are a few applications by now, but not much, eventually half a dozen, that they are already being used in medicine, let's say. But we have not carried out any study of applications. But as I mentioned, there are lots of papers coming up just studying them. And sorry, the expect improvement regarding to the, as I said in the beginning, the idea of making hybrid vesicles is just trying the best of two worlds. Not exactly pure polymer and not pure lipid. How far this has been succeeded, also I'm not on the area.
Yeah, I think we have to go on. Yeah.
Okay.
Yeah, sorry. Maybe more time for discussion at the coffee break. So let's thank the speaker again.
I would like to call Professor Vance from the Biochemistry Department of the University of Oxford to present us about the nature solar cells applying to nature technology.
Well, thank you very much indeed for the introduction. Am I the only person from the UK here? Yeah, I think I am. It's a delight to be in Moldova. I've never been before, and we've been looking forward to this trip, my wife and I, for a long time. We've had a great time so far, apart from the rain. So, I'm going to talk about nature's solar cells. Okay, so photoreceptors, in other words, ways of capturing light in nature, they are the most abundant proteins on the face of the Earth. You will know about chlorophylls, of course, but there is another very large group of photoreceptors in a wide range of systems, and this is an archaebacteria, originates about 3.5 billion years ago, and within the purple membrane of that archaebacteria is a protein called bacteriorhodopsin, and it humps protons across the membrane, and that provides sufficient energy for the cell to live. So there are a number of these have now been discovered. Bacteriorhodopsin, what I just discussed, is here. There are no salt beds, which this is in Moldova. I checked that. So this is a typical salt bed. This is San Francisco, but there are many around the world, of course. And it's a major way of getting salt. Yeah, I'd like the lights down if I can, please. That's much better. So, bacteriorhodopsin has been suggested for about a decade and a half as being a potential tool in bioelectronics, energy conversion, solar capture. The advantages are that it's got a very high quantum efficiency of converting light into energy. We need to learn this from biology, in nanotechnology. There's a wide range of absorption of visible light in particular. I'll show you some examples of tuning there. High thermal and photochemical stabilities. This protein is stable up to about 90 degrees centigrade. And they live in extreme environments, that's why they're called extremophiles. They have very large optical nonlinearities which can be exploited. Robust, you can put this protein into most kind of denaturants and it's still stable. Environmental friendliness and also genetic variations give enhanced properties. And I'll show you examples of that in a moment. Possible applications could be in solar energy conversion. Here's a potential cell. Those of you in those kind of environments who understand this. This kind of bacteriorhodopsin is put between two electrodes and then generates electricity from light. It's a sensitive solar cell. Obviously, reduction has been suggested because it clearly splits water into hydrogen, bioelectronics, carbon nanotubes, quantum dot hybrids have been produced, particularly in Asia, where they're studying this very extensively in places like Japan and Thailand and China. Image detection with tunable waveforms. So there's a lot of potential here for learning from nature. And that's quite important, and that's why I want to exploit that. We have done a little bit of this work. I'm now formally retired, but one of my side projects was to try and take this protein bacteriodopsin. Here is a bio-inspired detector that was established by our military in the UK. It's a 2x2 micron gold film, and we managed to get light triggering using atomic force microscopy in this system. And a number of papers on this and more kind of extensive work on that and using it. This has potential for about the same number of detecting elements as the 1-inch square detector at the back of your iPhone. So other photoreceptors, of course, and assuming you've not fallen asleep yet, are at the back of your eye. And these proteins too are called rhodopsins. We study bovine rhodopsin, but of course rhodopsin in the retinal membrane are extremely important for visual acuity. And here, one photon is incident on the rhodopsin in this molecule here in the rod cells. That produces about 10 to the 6 neural impulses in about 100 picoseconds with no noise. So it's a phenomenally good converter of optical signals into electrical signals. Just to show you that these have been around a while, but long after bacteriodopsin and the alpha-bacteria, they only started to evolve really about 700 to 400 million years ago. There's a big discussion about whether this is convergent, divergent evolution. So why did I put this up? Because a potential device based on this kind of technology, but not from my lab, from Bob Burge's lab in Boston, has been implanted into the back of human eyes to give sight to people with retinal pigmentosis. So if you're blind, you give them some visual awareness. So in all these systems, we're simply taking water and breaking it down to its constituent components. But are there microbial rhodopsins that have an even higher quantum efficiency or faster kinetics than bacteriodopsin, which has been studied so extensively? And we found one of those. And then, if it is, how is any increase achieved at the structural or molecular level? So here's a phylogenetic tree of many of these archaeodopsins, and the one we've chosen is a group of 4 of them, archaeodopsin 1, 2, 3, 4, and we're studying 3. So the whole crux of this is the water, and this is going to be my main message. And of course, we all know about the requirement for water, all sorts of molecular interactions within the cell, and what can we say about the nature of the water in biomolecules? And this, I think, is going to be generic with all biomolecular systems that we study. In other words, it's applicable to many systems. Now, the link between these archaeodopsins and the rhodopsins in the back of our eye, and all of them with this molecule, is retinal vitamin A. About 200,000 children are born every year with vitamin A deficiency and are blind for life. And it was discovered by a chemist in Leeds, George Wald, in 1939, for which he was given the Nobel Prize. Retinal, small molecule vitamin A, at the center of these proteins is triggered, and the light triggering of the rhodopsin, of the retinal within the rhodopsins, that is what splits the water between into its constituent parts, and it's covalently attached through a shift base to lysine in the protein. In the early days, Richard Henderson from the UK managed to get low-level, low-resolution structures at about 7 angstroms using electron diffraction. That has got better over years with better techniques, and now the rhodopsin, the retinal within the protein, can be resolved to a reasonably good level. Our solid-state NMR work, which I'm not going to talk about, resolved it to a much higher level, way ahead of crystallography, and that's all published and available in the literature. So what is going to happen here is that light is incident on the retinal, either 13 cis or trans, and then that takes a proton from one side to the other side, so proton transportation through the protein is how this works. This is the protein release complex, and converts the water into a proton and a hydroxide. So we do this across the membrane like this, and that generates about 150 millivolts membrane potential, which is quite standard in biology. You'll hear more about this from Jim Rockman tomorrow maybe. And so this is quite a high voltage. Now if you work this out, this is 150 millivolts across about 4 to 6 nanometers, and that's giving you a potential gradient of about 15 kilovolts per centimeter. I challenge any electrical engineer to produce a capacitor that does not break down that kind of voltage. So far, in biology, it happens all the time. So this membrane potential can now fuel the cell without any other source of energy. Now the solar power here, about 60% photon capture efficiency. Now, solar cells that we see all over the place, I've seen them on my travels over here in Eastern Europe, they capture about 20% of the light capture efficiently. I think in the Fraunhofer Institute they've got up to about 49%. Of course, we want to increase that yet further, and you may have more information if you're in the field. So this archaeodoxin 3, this protein I'm going to talk about, is a proton pump similar to bacterodoxin, but it is 10 times more efficient. And that's been observed by us and by others now. It has a very similar sequence homology to the bacterodoxin we're talking about, and the other of the proteins in this kind of series. Has an extra chromophore here, and that produces a bright red color, so we can produce lots and lots of these membranes in the laboratory, and it grows very easily indeed. It's from Sodomensis, so it's halorubum, and it comes initially, it was detected in Tibetan salt plates at high altitude. And it's almost certainly evolved to produce this higher efficiency because it's in a colder climate than the bacterodoxin, the archaeodoxins and so on, which are in a much warmer environment. So the oligomeric structure was not known when we started this project, and the structure of the protein was not known at all, because nobody could crystallize it. So here is the photocurrent generated for AR3, and here for bacterodoxin. You can see the difference in the efficiency of this protein. It really is much more efficient. Now, why would we bother to study yet one more of these proteins? Well, the reason is that, it's used in about 1,000 labs worldwide for optogenetics. This is transfected into neural slices or in live tissue, and that should be a movie, sorry, transferring it to a PC is not working. Could you just click the image, that image in the center, and see if it works? You can then see the triggering of the action potentials along the neurons, and this is a rat hippocampal slice. Anyway, you can't see it here, but no, okay, sorry, it was a movie that works, but it's not working here. I suspect a Nobel Prize will go for this field fairly soon. So what we have done is we put this into a double binary between vesicles, much like Manuel was talking a minute ago. We put the protein between these, and now we can put an electrode in, and we can measure the change in voltage, photocurrent as we irradiate with light. So we can show that we have an active protein driving a proton gradient between these two compartments. This is the same technology that Hagen, Bailey, and Oxford developed, and now was put into a company called Oxford Nanopore, and was floated on the stock market for $4 billion, and is used now for sequencing hemolysin and many other macromolecules. So if we now look at the photocurrent generated in the absence of protein as the green line, bacterodoxin is the purple line here. I'm sorry, because of the lighting and so on. This is where the light is switched on. It's a much faster rise for our protein AR3, and then it drops off, and then as the light switches off, the photocurrent stops. So we can repeatedly switch on and off the photocurrent, and here's another second pulse in the system. So in our system, this droplet binary interface, droplet interface binary, we have about a two-fold increase in the protein activity. And there's the power spectrum for you, showing a different spectral response. So that can be done repeatedly, just on, off, on, off, on, off, on, off. And so it's a nice kind of photo switch. Now, if we want to understand how this protein works, we need structure, and we need it at the atomic level. I've shown you some crystallography from bacteriodopsin, and when this protein was brought to us, in fact from Shanghai, I brought it back to England in my pocket through customs. Nobody said any words about the culture, but it grew extremely well in my lab. And we managed to crystallize it with a student from Japan, in fact. And so we crystallized it using the lipidic cubic phase, which everybody knows about for membrane proteins, but with no detergent. So this is a first. We're not putting this protein in detergent and then putting it into crystals. We're doing it directly into lipid. So there's been no denaturation of the protein. We use the conventional methodologies. We can change the conditions. We can get very large crystals, or in this case, very small crystals, and I'll come back to that in a moment. And we got pretty good resolution. We can use molecular replacement, of course. And in the ground state, we have a structure of 1.07 angstroms for this protein. And that is a long, big advance on the 7.5 angstroms in the early days, and it's now approaching a pretty good limit. There are about 15 structures in the protein database now, and we've got unusually also some dark adapted structures. The structures initially are here in this Nature paper. We also took this protein. I'm from a biochemistry laboratory, so we like to know what our protein is, we like to know what the components are, we like to know it's functionally active. We've shown that already. And we took the protein from here into a lipid, a styrene maleic acid polymer, much like the polymers that Manuel was talking about, and put it into a lipid disc. These discs are quite important. If you've had a COVID vaccine from the big Moderna and Pfizer companies, this is the system that the mRNA was in. We have delivered chemotherapy drugs with the medical group in Oslo to mice. So it's a good drug delivery vehicle. There's a lot of technology involved here. And then we did electrospray mass spectrometry, and we've now shown that we have certainly the protein there, which is here, AR3 with the lipids, and some of the endogenous lipids, because we had to add back some lipids into the system. What's important about this is that this is at low voltage. A membrane protein normally would give a big hump, and you have to deconvolute the data. This is behaving like a soluble protein because of this. That has generated a new company in Oxford, Spin-off, which is now functioning at the high level, looking at drug receptor interactions, in particular GPCRs, using this technology exactly. So a lot of technology is spinning out. Now, the retinal I showed you, now we can see, in fact, that the retinal is a very high resolution because, of course, it's residual electron density. You normally get, and you then model back the retinal. We use NMR parameters in the early days, which is independent of the electron density. And now we can see why the protein is a different color. There are more residues in the binding site, about 5 more, and that's why you've got this shift to the red from purple over bacteriodopsin. We can resolve the all-trans isomer, and also something we showed 25 years ago, the ring actually has got 2 orientations within the protein. And that is something that Martin showed, but crystallography couldn't at that time. Also interestingly, this retinal, in the dark adapted state, this is no light whatsoever, is actually in equilibrium between the 13 cis and the all-trans. And that's quite unusual, that hadn't been expected. So there's obviously some interesting energetics going on there. Then we did some potential mean force calculations with this, with a group in Israel, Igor Shapira, and we found out from these calculations exactly how that happens. the energy difference in the dark adapted, that's the blue, is quite small between the cis and the trans. So you should expect thermal equilibrium, you get a population of both energy levels. But in the light adapted form, the energy difference is much higher. That's why it's all in the all cis in the light adapted form. And that has been an enigma and hasn't been thought about before. Now I talked about spectral tuning. Now most man-made detectors are either switched on or off, and they usually aren't sensitive to different wavelengths. They use kind of filters and Bayesian algorithms to put that information in. But in nature, we detect color. And it was suggested about 10 years ago that if you could change the electrostatic potential around the retinal within one of these proteins, this is theoretical paper, totally theoretical, then you should be able to change the color of one of these proteins. Now we can test it. So we have done a wide range of mutations of the residues around the binding site to get bathochromic shifts. In other words, we increase the hydrophobicity, and then we get a red shift in the protein, and then a hypsochromic shift, and we get to change the electrostatic environment, and then we get a blue shift. Multiple variations, and the student in the lab did about 23 of these. Here are the absorption maxima for many of these. You can see the membrane now is not red anymore. We can make yellow, purple, green, pink, just by changing residues in the protein by mutation. Here's the mutation library, and we've got some redshifted ones there. So, just to show that the blue one, for example, and I'll tell you why blue is important at the moment, is also shifted. That one is active. So here's the photocurrent output as a function of light, and we can switch it on and off. Here are the two membranes, one blue, one red. So we can tune the wavelength of this. So if we're going to use the detector, we have two different wavelength sensitivities. Now why is that important? Well, in optogenetics, if you had two different colored outputs, you could then do multiplex voltage imaging into the system. So you can now detect two different effects within the same neural synapse. And that would distinguish the activity of different excitatory and inhibitory neutrons in intact tissue. I'm now aware that people in this field are using this kind of approach as a result of this work. Now, why is this protein more efficient? So it's more efficient. Now, here we have the proton pathway through the protein. I showed you a cartoon earlier. Here are the residues that are involved, the important residues that are involved within that splitting of water into a proton and hydroxyl ion. It's called the proton release complex. So we're going to focus in particular on this arginine, and we're going to compare it with bacteriodopsin. So this is that arginine in our AR3 in a dark adapted and light adapted structures. And that arginine 82, now we have such high resolution that the only way we could fit it is into three different orientations, three different rotamers. So it's got three different positions around the arginine side group. And that implies to us immediately that because this is the most important residue in the proton release complex, that must affect the hydrogen bonding within this system. And sure enough, we could model that in. And compared to the electron, the kind of receptor, etc., and donor residues in here, here's the arginine, then we can see there are much weaker hydrogen bonds, 2.6, 3.1. These are the three different rotamers in the protein, multiple hydrogen bond networks, so multiple pathways for the deprotonation across the proton release complex. Multiple pathways for the proton to grow through the protein. In fact, three of them. So if we compare that with bacteriodopsin, which is here on this side, there's only one pathway and much stronger hydrogen bonds, 2.3 to 2.6. So this is, if you want to imagine this as a highway, you've got a single highway through the protein of bacteriodopsin. For AR3, we've got three. So you've got three channels you can go through, three potential channels. So that's one reason why it's probably more efficient. So the other reason is that there's an omega loop in these arginine. They have evolved to produce a loop. Can you see the omega here? We have something from Greece here. That's an omega, right? So
Yes.
it's very clear. Whereas in Bacteroidopsin in purple, that omega loop does not appear. Now what does that do? When I looked at the electron density around this region in the protein, here it is, and then you look at the water molecules, there's a huge reservoir of water retention here in this protein. So there's a big tank of water ready to be deprotonated and formed into ions across the protein of AR3, which does not exist in Bacteroidopsin. So that's another reason why AR3 is more efficient. It's got this water reservoir ready for translocation across the proton-release complex. Now finally, we wanted to do some kinetics on this process. If it doesn't move, it doesn't do anything. And in biology, we're always interested in dynamics. And there's a methodology called free electron laser, X-FEL, where we can now irradiate a crystal at very high intensity and very quickly, so we can get an instant picture of the molecule atomic form without any damage. And Soiwata at Spring 8 in Japan has set up a really beautiful system where we can irradiate with a laser pulse onto our protein. So this is why we needed to make nanocrystals I showed you in the early days. They are fed in front of the X-FEL's beam, and then when we use the laser pulse to irradiate the protein with light, and light activated it, we shoot it with the free electron laser to get the structure. So we can get structures now at different time scales. And the way this works is here. Now, if you suffer from strobe lighting, don't look at this. I'm going to show you. Here we are. Here are the crystals coming through the Hamilton syringe, which is where they're produced from, the end of the syringe. And here's a little kind of a funnel that sucks away the crystals. And this is the laser light going through the protein. So it's a very interesting, very nice setup. It's beautifully set up. I could sit in front of this, and it takes about 20 minutes to empty a Hamilton syringe of crystals. We could take those to Japan, of course, and they gave us 3 sessions, which was great. And as somebody who knows nothing about it, I could do different electron density profiles across this protein as the data was coming out. We now have structures from femtoseconds down to milliseconds. So the difference map at 5 femtoseconds, just one of the 15 time points here, shows that there is a change in electron density around the D85 in the protein, here's the retinal, at this time scale. So already at 5 femtoseconds, water is shifting and moving within this protein as part of the functional cycle, which is quite nice to see. There are movies for Bacteroidopsin out there in the literature. Other people have done much more than us on this. But I think there's a different construction. So the protein is actually active in the crystal, which is also nice to see. It's never seen detergent, it's only been in lipids. So if I could conclude water plays a vital role in protein here, photoreceptor action. And if you're going to use something like Bacteroidopsin or other biomolecules to have bio-inspired detectors or devices, you need water. You can't get rid of it. The higher efficiency for this particular protein is a way to manage at the molecular scale to give it faster kinetics and to give it a much better photon efficiency, photon capture efficiency. The data is showing initial insights into water movements. Any device, bio-inspired device, must include hydration. And there are generic implications, and I think this is going to be important as our methodologies get better. Every biomolecule needs water, we know that, and Gorgi said that. To understand it at the absolutely atomic and molecular level is going to be vital. So just to acknowledge the people who did the work. Next slide. Yep, okay, so Carol Robinson and Hagen Bailey involved in some of the activity measurements. Peter Judge drove the project, senior postdoc in my lab. One is in the EPFL, one Javier is at MIT. Isabel from the National Physics Laboratory drove the crystallization. This is us down at Spring 8. Diamond, of course, the synchrotron in Oxford. I didn't show any of the high-speed AFM on these systems, but we've done that as well with Manuel in Montpellier, but also with Miki Subate in Kanazawa University using Professor Ando's laboratory, and then Igor Shapiro for the calculations. So, thank you very much.
Thank you very much. A quick question, if there is any.
Thank you very much for the very interesting presentation, and it is definitely a very promising direction, and optogenetics gives us a lot of hope for in the future pigmentosa, and therefore, of course, such diagnostic tools about which you are talking, they are very interesting, very promising. I would like to ask you, how did you check, how did you prove the efficiency of or the wanted, the desired efficiency of the mutations that you induced in AR3 in order to get them the outcome regarding the current change?
Sure. So there were a large number made, but only 20 or 3 of those were the ones that we saw were active. So we assumed that those active ones were correctly folded, really. So that's a good indication the proteins working and functioning, but they're all sent off to sequencing. All of these proteins were routinely sequenced, every one of them. So and of course, I didn't have time to tell you, these are expressed in E. coli, they're put back into the system, and then retinol is added back to the protein to form the shift base for the lysine. You can't do this obviously in the original organism, but you can clearly do it with the plasmid. So that's the way we did that. I just didn't have time. All the details are in that Nature Comms paper, and also there's an analysis paper there and something else as well. When we get to write up all the other structures, if we ever do, then it'll be there too. But it's an important question. We do all the controls, and we never study anything in my lab unless it's functionally active, which makes graduate students cross, because there's so much structural biology on systems which have no functional consequence. But that's my own philosophy.
Thank you.
Thank you for the question.
Okay, thank you very much.
Right.
The next speaker is Max Catalan and he will present about bionic limbs and post-amputation pain. Here.
Yeah, no.
Thank you very much for the invitation. My name is Max Catalan. I did most of my work in Sweden, where I spent about 16 years, then I was in Australia for a while, and last year I decided to retire from academia and do humanitarian work in Ukraine based on the research that I developed during my research time. So I work with prosthetics. The challenge that we face is that we want to replace a biological system with an artificial one. And when it comes to prosthetics, there are basically 4 challenges: the hardware, the robotic part, the way the person will control it. If we want to have dexterous control, we need to have some sort of sensory feedback, and all of that is not very useful if we cannot have a way to attach it to the body, because otherwise patient won't be able to use it. So the prosthetic hardware is quite advanced. We don't really are taking all the juice out of it. Where we struggle is in the way to control it. And to control it, we need 2 things. We need to be able to extract motor information from the body and make sense of it. And for that, we can decide that we're doing that from the brains, from the nerves, from the muscles, what kind of surgeries are we doing. We're using electrodes or optogenetics particularly. That's something that we investigated now. And based on that, we have different electronics. Maybe I'll get close to this in case I close and you can do a PhD basically in any of these topics. So one way of doing it is placing electrodes on the surface of the skin. As you can see there are some electrodes in my forearms. If I will use my hand, I could still, in principle, control every finger and do it simultaneously using the coders. And we have done this, all the algorithms we've done it open source since 2013 actually. So if you go to that website, you will see all the code, the electronics that we use that can be used to control a prosthetic device or an exoskeleton. But the problem is that algorithms will only take you so far. If you don't have good quality and stable signals, they won't do miracles. So what we can do is look at the source of information, and we know for decades that if you implant an electrode, you're going to get much better signals. The problem is that once you go inside the body, you have to communicate with the prosthesis, and there's a skin interface, and you can not just have a wire going through. So the challenge is that you need to have something that is safe and stable, and the bandwidth tends to be one of the issues with prosthetics. You can do a wireless solution, and there are different ways of doing that. You can have a centralized system or a distributed system, but so far that has not been implemented in long term in humans, with the exception of one experiment. So we went wired, and the reason why we went wired is because we had another machine interface, which is the way you attach the prosthesis to the body traditionally is with a socket. And what a socket will do is compress the soft tissue very strongly to hold the weight of the prosthesis. And this is actually one of the sources of a major problem for people with amputation, because as you can see the mark of the socket, and also when you have a fully functional joint that cannot be used because the technology that we're using to attach the prosthesis. So that limits even further the functionality of the patients. An alternative to that is to use osseointegration, so you put a titanium implant inside the bone, and then you basically do a skeletal attachment to the bone where you can attach the prosthesis. This was discovered in Sweden in Gothenburg in the 50s, and there were dental implants, and it was used for skeletal attachment of prosthesis in the 90s. So when I joined the field, osseointegration was there, and what I've been working on is developing a system in which we can implant electrodes in nerves and muscles, and then re-engineer the osseointegrated implants so we can get those connections out, analogous to a USB connection to the human body, the nervous system. Could you hear that in the back? Well, he's basically saying that it's great. Now, so the big difference is that now the patient can wear the prosthesis the whole day. He doesn't have, many patients when they get home, they take off the prosthesis because it's uncomfortable. In a similar way that you would never sleep with your shoes on because it's uncomfortable, having a socket on your skin is uncomfortable. Particularly patients with the lower limbs, they often will think about, okay, if I have to stand up and go to the kitchen and go there, they'll plan so they don't move around, because when you're walking in a socket, you have a bit of pistoning, and that can be uncomfortable for the skin. So, with my first PhD student, some time ago, we developed an embedded system so we can also provide sensory feedback, and what we achieved is having a system in which electrodes implanted in the body are used to control the prosthesis. There's a sensor in the thumb of the prosthesis. When that sensor makes a contact with an object, it can stimulate the afferent nerves and provide sensations to the patient. So the patient is effectively feeling there is an object on his hand. And not only there is an object, we can modulate the stimulation so the sensations of touch is graded. So the pressure that is placed on the object can be transmitted to the patient. So this was the first time that a patient could use implanted electrodes for control and sensory feedback safely for many years. So the first surgery was done in 2013, so it's more than 10 years, almost 11 years now, where the patient has been using it in daily life. But the control is very rough, so the patient can only open and close the hand and have one sensation. And what we try to do is find ways to increase the control. We started looking at working the problem from the biological side, because we were a bit stuck on the engineering side. So what you can do is basically rewire the nervous system, because many of these muscles don't have biological functions. The biceps and the triceps in an elbow amputation are not actuating any elbow. So you don't need the force of those muscles, but you can use them as biological amplifiers. So what you can do is take one of the nerves that used to go to the hand and reconnect it to a muscle that has no biomechanical function. And in that way, what we do in this case is that the biceps, it has two heads. So you take the musculocutaneous nerve that goes to one head, you cut it. You take the median, in this case was the ulnar nerve that goes to the two last fingers. You transfer it there, so when the patient thinks about closing the hand, the short head of the biceps contracts. When the patient thinks about flexing the elbow, the long head of the bicep contracts. So now we have two signals instead of one, and you can do the same in the triceps, and that allows you more function. So we've done this with a few patients. They have used it for, that report was 3 years in activities of the daily life, receiving sensations and controlling the prosthesis. But again, that's not as many joints as we have in a human hand. So we look at this again, and we decided to get more creative on the surgical reconstruction. So instead of only passing the whole nerve to a muscle, you can get the muscle, split it into different fascicles, get one of the fascicles to a native muscle, and then the other ones have a free muscle graft, say from the leg, connected to those fascicles there. I'm running out of hands here. So here, so one fascicle goes to a native muscle, and the other ones are connected to these small pieces of muscles in a Prieto manner almost. So when the patient tries to do movements, all these neuromuscular contracts contract, and then we use machine learning to decode what the patient is trying to do. And then what we can have is intuitive control. So this patient is thinking about flexing and extending the elbow, rotating the wrist, and actuating that terminal device that he prefers more than a hand. In the same way he will do it with a biological hand. And that's quite cool, but what we managed to achieve in this case is to have a patient with a above elbow amputation controlling the flexion and extension of every finger. And again, this was the first patient, not the only patient actually in the world that can do that, because we haven't done more surgeries after that one. And it's all possible because a combination of technologies. There's a novel reconstruction procedures. We have implanted electrodes to extract the information, get them through the osteointegrated implant that we also use to connect the prosthesis, and then we use machine learning to know what the patient is trying to do. We have done it in a lower limb, well actually done it in lower limb as well, but this is in a below elbow amputation. In this case, the patient has a hand that has sensors in the thumb and the index finger, so she can feel how squeezy the different objects are. So some are softer than the others, and sort them out accordingly. And then we start running into challenge because we're having a lot of hardware inside the body, and the risk of infection increases the more foreign materials you have. So that starts to be a bottleneck. Another problem, a lot of my work has been problem driven, and working with people with amputations, phantom limb pain turned out to be, for many of them, even a bigger problem than not having an arm or a leg. Because if you're in pain, and I'm sure you have been in pain in your lives, you can do anything. It's hard to concentrate, it's hard to wear a prosthesis, study, work, it has a high impact on quality of life. So we know that phantom limb pain is quite common. Most people with amputations will feel it. If it is not treated properly, it can become chronic, so it's harder to treat. And as I said before, because it demands attention, we know that it has a negative effect on quality of life, employment, education, social prospects are diminished when you have pain. And I'll touch on depression a bit later. So we struggling to understand phantom limb pain. Acute pain is quite straightforward. Neuropathic pain becomes more complicated. Phantom limb pain can have sources on the periphery or centrally. For the peripheral sources, these surgical procedures that we've been working on, they seem to be very effective for neuronal pain. So now we're doing a large international clinical trial in North America, Europe, and Australia, where we are trying to produce high-quality evidence of the effectiveness of these treatments so that we can help to shape policy. So in the beginning of my career, I was very focused on doing cool things. After that, on going through the breakthroughs of the knowledge that we have, now I'm focusing on getting those technologies that we know work widely spread. And to do that, you have to start working with governments and policy, creating data that is considered good enough for them to change the way they've been working so far. We develop step-by-step instructions on how to do these procedures. These are open, freely available videos on how to do that. The prosthetics that we develop are quite good at reducing phantom limb pain. It kind of makes sense. The problem is that they're extremely expensive. The cost of doing any of these prostheses is about €1 million. We don't a handful of patients around the world. It will likely never be in a developing country, right? So, growing frustrated with the low accessibility of those technologies, I developed a treatment that is much lower cost. If you want, you basically need a computer screen, and a webcam. And with that, what we do is, using similar technology, we put electrodes in the surface of the skin, and then in the image we show the patients a virtual limb. So this is augmented reality, similarly what the mirror therapy would be. The main difference is that in this case, for that virtual limb to move, the patient has to do an actual execution of the movement. Because muscular contraction is the ultimate response to when you attempt to do a movement of motor execution. Now, the therapy is not based on looking at your limb, it's based on doing phantom exercises. And if we want to increase the volume of the exercises with the phantom, then we can employ games. And in this case, if you will see this missing hand, you will see that when he rotates the wrist to the left, the car goes left. When he rotates to the right, the car goes right. He's flexing the elbow to accelerate and extending for braking. So the volume of exercise increases considerably. If you're training with neurotherapy, you can do maybe 20 repetitions. If you play 10 minutes again, you will do 100. And this is what we think is the reason why this treatment is more effective. So the first larger trial of this treatment was done with patients with chronic pain. They had, in average, 10 years phantom limb pain, and they had tried everything that was available in their clinics. So medical options and also non-medicals. So that's the intractable part. And what we found is that in only 12 sessions, we could reduce the pain in about 50%, and that stayed for about 6 months after the last session. And another effect that we knew from our previous case studies was that the reduction on interference that pain had in their life was considerable, particularly sleep. Many of these patients cannot spend a full night of good sleep because they'll have an episode of pain that will wake them up. And if you don't sleep well, your pain threshold goes down. And if your pain threshold goes down, you don't sleep well. And then you enter in this negative cycle that is kind of hard to get out. So because this technology is much more accessible, I managed to distribute it in many countries, but in a few clinics. So there's been a few hundred patients treated with this technology. Then going into the science of why this works, it seems to me that the reason is that we basically engage in the circuits that were affected by the amputation, and there's, like I talked an hour about the hypothesis that I proposed for the pathogenesis of phantom limb pain. But if we're aiming to engage the circuitry that was affected by the amputation, then it makes sense that we also try to engage the sensory part. So we developed this system that provides sensory and motor training, and that's what we're doing in Ukraine. You are aware there's a war. It's been more than, I think, 150,000, the last estimation I heard in the past 3 years. Research with people with amputations is kind of tricky in any country because there's not that many. That's why you normally have to do clinical trials that are international. In Ukraine, there are a lot of people. So in half a day, the first time I visited Ukraine in early 23, in half a day I saw more people with amputations than in my whole career working with prosthetics. So it was very clear that the need was much higher there. So what we've been doing is implementing these technologies, as I said before. That's a patient with an upper limb amputation, controlling a hand. We also done it for lower limbs. We have similar results. But one problem is that war injuries are not simple ones. So I was used to working with people in rehab centers where they had the amputation, but everything else was fine. War injuries, they have large defects, they have burns, they have injuries that they last months. They'll spend 5, 6, 8 months in a hospital bed, very often not getting any rehabilitation. And at that stage, they develop phantom limb pain, and the opioids that they receive are not enough to treat it. So following research that we've done, basic research on phantom limb pain, we developed a treatment that is implemented in an app that doesn't require the electrodes. And so far we've seen similar results. You can read the subtitles. So this patient was not able to use the standard treatment because the amputation, you can see the amputation is very high. So you have a shoulder disarticulation, a hip disarticulation, there is no need to put electrodes on. And also if you have scar tissue or burn, you can record EMG signals, so you cannot treat it with the standard treatment. A couple of months ago, we published an article, and this is only one story of the many patients that have been treated with this treatment. So this was a patient in the Netherlands. In the Netherlands and Switzerland, as you know, euthanasia is legal, and the most popular reason to request euthanasia is chronic pain. So normally to be accepted in the process of euthanasia, you're evaluated by psychologists, psychiatrists, and so on. It's a long process. And after that, they give you the go, and then they can help you to terminate your life. And we had a patient who was already accepted for going through the procedure and enter one of our clinical trials based on the technology that is not using the electrodes, but the app. And he got better, and he called off the euthanasia. So that's just an example of the effect that pain can have in someone's life. To the point that when the depression and the suffering is too much, they want to finish it. Now the issue with phantom limb pain is that it's not very well understood, and because of that, it creates a lot of stigma. I have spoken about this in many countries, so I remember giving a talk in China, and done this in Latin America, and sometimes I'll have doctors that will come up to me at the end and they will say, oh, that phantom limb pain thing is interesting, my patients don't have it. And I hear this in a few countries and I go, well, that's weird, it seems like it's only white people having phantom limb pain. But it's not really that. It's cultural differences and the way the healthcare is provided. So many patients will be afraid to talk about their pain because they're afraid that other people will think that they're crazy, because how can you have pain in a part of the body that is no longer there? It's not properly how to treat phantom limb pain, or phantom limb pain itself is not properly taught in many medical programs, if it's taught at all. So we met many patients that their doctors told them, no, it's in your head, you'll get better, so they dismiss them, they get no treatment, patients don't go and have treatment. So this is a large problem, and it might look like. So the education is outdated with good reasons, because we just learned more about phantom limb pain in the recent years, and that leave a lot of patients without treatment. So we just launched a global, you can say, educational campaign on phantom limb pain, where there is a website where we provide up-to-date science-based information, because there's a lot of snake oil of sorts. When you're in pain, when you have a medical condition, when you go to the doctors and the doctors cannot give you a solution, particularly if it's pain, people try all kind of stuff, and some of them are dangerous. And unfortunately, there's people that are happy to make money out of the suffering of other people. And that's something that, you know, I personally don't like, and I guess many people don't like, so we try to provide debunk treatments that are just snake oil, basically. So we have, we try to educate through the modern times, so using social media. We have the main information on the webpage, we have social media, and then we have short snips of content. We develop some animations trying to explain how to do more effectively mirror therapy, not necessarily all our treatments, surgical procedures. We organize courses on how to do the surgical procedures, particularly in Ukraine, but we have one in Africa and in Asia coming up. And then we have people, you know, describing things. And we learned that in some countries it's important to describe this in their language. So we have included a few languages now, but we just launched Arabic, again, because there's a high population of it's a lot of people that speak Arabic that don't speak English. So we're probably not going to bother to put it in Dutch because, you know, everyone in the Netherlands speaks English. But in these other countries in Latin America, it's not that common. So all this is a volunteer work. So the animations, we have a group of a dozen of volunteers from different countries helping us to run this initiative. And yeah, so just to say, these technologies, we have plenty of clinical studies and scientific papers on phantom limb pain. We have used it for stroke, chronic stroke, as well as hand replantation and transplantations. So through the 10 years or so that I had of academic work, we published over 100-something papers, scientific papers in different journals. But now, as I said, what I'm trying to do is get that technology to really benefit the people that was intended to benefit when I started doing it. So now I do voluntary humanitarian work in a few places in Ukraine. This is one of them. It's a pain center where we primarily see people with phantom limb pain. And I also work with a center in Kyiv where we actually provide as good prosthetic fitting as anywhere in the world, all done by donations. So we have osseointegration, pattern recognition systems with my electric prosthesis, power elbows, hands, and so on. Unfortunately, it's getting harder and harder to raise funding to provide prosthesis to these patients. Another foundation I'm working with, it's called Remedy. We try to help students that lost their family in the war to continue their education. So these are talented students that have good grades, but now they're struggling to, you know, continue living, and if they don't have some funding, they'll have to quit the job. I had a scholarship myself, and that's how I got my education, so it's kind of close to my heart. And what we do is we try to find mentors from around the world. Say, if it's a professor of biology in Europe or the US, we get a student that is doing biology, they can guide them and support them with about 100 euros a month, which is not much for a salary of a professor in Europe or the US, but it makes a huge difference for these students to be able to finish their education. So if you'd like to join us with that, all the information is on our website. The money doesn't come to us, it goes directly to the students. So that's some of my work that's been more practical. We have a few websites and so on. You're welcome to ask some questions right now. We'll have some in a minute. Or otherwise you can contact me online. Thank you.
Thank you very much for the work you do, and I'm sure lots of people appreciate having mobility. I think they would appreciate that, especially for war in Ukraine and Russia. I have two questions. For neuromuscular training, are there exoskeleton or simply by exercise or by training?
So it's quite interesting because when engineers got involved in medicine, particularly on robotics, exoskeletons seem very promising, but so far they have not shown to be much better than physical therapy. And it really comes down to the volume of exercise. So if we all know of an athlete that they were very good and they had an injury, and the doctor told them, oh, you will never walk again. And they're running thereafter and winning competitions. They do that because they're highly driven individuals. So somebody that's very motivated, they will train, they'll get better, and they'll overcome the disability. The problem is that that's a minority of the population. Most people, you have an injury, you stop trying to move, you have this learned paralysis, you really don't do the exercises. And then what it seems to me is happening is that some technology can help to get those people to a volume that will get them to be better. So exoskeletons can help on that. And we know that if you are sending motor commands and you time that with electrical stimulation together with physical movement, will be a much better rehabilitation than if you try to do it without any of that. So the technological solutions for rehabilitation, they can work, but we have to be very mindful of the physiological process that go together with it, and not only just because it's a nice looking robot.
And the second part is, you mentioned about the traumatic injury. Does that also include traumatic brain injury? Because in that case, having a training perhaps would be a little bit more challenging, and that's where exoskeleton perhaps might help. So do you include TBI in your research work?
So we haven't included that, but at the end of the day, there's a problem with the brain. So the reason why there is an issue, either because it was a stroke or a traumatic injury or anything else, you still have compromised circuits. And then what you try to do is get the rest of the brain to compensate for the compromised part that is affected for whatever reason. So what we've done in a stroke, for instance, and I didn't show that for the lack of time, we have patients, in a stroke you will get most of your recovery in the first 6 months. And after that, there's very little that you can do. So in many places, after 6 months or a year, they don't even give you therapy. People just stop with therapy because they gain very little. And we did a trial with this technology, patients with 1 year stroke after 1 year and a half, and we found that they could regain hand function. That's clinically relevant, so still grab and let go an object. So all this to say that the technology that we're using is for high impairments. You try to move and nothing is happening, so that we can pick it up from the muscles and then allow it. Exoskeleton will be the next step. We actually had a patient that had an arm replantation. So he had an accident, his arm was cut off, he took the arm to the hospital, they connected again, but not all the function was recovered. And he didn't have function of the thumb. So with the system, we get them to move the thumb enough so he can use a glove, which will be a soft skeleton with a sensor. So when he grasps an object, the sensor makes contacts, and then with pneumatics, then he can have a full grasp. So not all technologies are good for every stage. Our technology is good for the beginning, the high environment, and then when you can do a bit of movement, then exoskeletons are probably something. So also the technology has to adapt based on the environment.
Thank you so much.
Okay, thank you very much for your talk once again.
The European Biophysics Union wants to make an announcement in the Congress, so Professor Manuel.
Thank you very much. So the first time I met Theon was at the International Science Council in Paris, and at that point he contacted me and invited me to come along. And it was clear to me that Moldova is one of the countries which is not currently a member of the International Union of Pure and Applied Biophysics. And so it struck me as a good opportunity to come and talk to you and your community. Now, can you go next slide? No. Okay. So IUPAB is a world federation of organizations in biophysics. And that's all encompassing. It's pure and applied. So it includes medical biophysics as well. And we've been hearing some of that here, in particular in Eastern Europe. Founded in 1961, which promotes biophysical research and education. Young people can benefit from this. I know you're leaving, but young people can get travel bursaries through IUPAB to our Congress, and I think we should go. So just keep that in mind. Adhering bodies and observers from 60 countries around the world, about 12,000 members. We have national biophysical societies, regional ones, national science research councils, and academies of science, scientific academy here. 3 regional organizations of biophysics are affiliated also. One including Asia, which is all the way from India through to Australia. The European Biophysics Society Association. Now, with your vote recently to move more towards Europe, it would be appropriate for you to join that. And the Latin American Federation of Biophysical Societies. There were changes in 2024. We had the Koreans join after 10 years away, and a new Vietnamese society was admitted in 2024. And we have new observers. In 2021, there was nothing in Africa. Now over 14 interested groups have joined a Society for African Biophysics. The Mexican Society has been founded, and now Singapore. So we are, this is the European group, one of our groups. Those countries that are shaded in lighter blue are members, and you can see, of course, that Moldova is not a member at the moment. But countries around you are, and Ukraine is indeed an observer. And with, as we heard from the Japanese ambassador yesterday, the rebuilding of Ukraine that will come, you obviously could be part of that benefit as well. We at IUPAB and EBSA would like to be part of that rebuilding of the community and interaction between them. So the other aspect for Moldova that's important is that your interaction with the European community, our activities and our congresses, which happen every 2 years for EBSA and 3 years for IUPAB, give you that interaction to collaborate, to find collaborators, to go to the European community, and we have representatives that go to the EEC in Brussels for funding and collaborative grants. And for student mobility, and we heard that again yesterday, you've got this bulge of people within that 35 to 45 age group. Mobility from Moldova to other places. Many of the scientists talking here have clearly benefited from that mobility over their careers. So join up with the European group, it would be great. You can be an observer to begin with, but it's only €2 per member for EBSA. And both Manuel, who is here at the front, and me, we've both been presidents of EBSA, and now we're presidents of IUPAB. Manuel's the past one, I'm the present one. The next Congress for IUPAB and EBSA will be a joint Congress, and it will be in Berlin, which is close by. So it's very easy for you to come from Moldova, and it would be great to support some young Moldovan scientists in this. And just quickly, we've joined the light sources. If you don't have light sources for crystallography and access, you can get support through IUPAB to go to those. We're part of the Earth Humanity Coalition, which is now very important, of course, and we're members of the International Science Council, which makes science policy up to the UN and to government. So we have input into all of those. And these are the members, the Secretary of Australia, here's Manuel, he's here, I'm here, next President Andrew Brundle, and we have the Treasurer. And then we have Young Investigator Awards. We have send people from less developed countries to the Biophysical Society. We fund their travel. We fund travel grants to students and plenary lecturers. We could have funded something for this if we'd been appropriate, you'll remember, for example. And then in 2025, a number of events that we funded, including this satellite And we spend about 70 to 100,000 euros a year on these activities. And here are some more of these travel grants. So we're supporting younger people in particular to come to these. There's the Berlin Congress. South Africa, we're funding a session at this big meeting, and then Quebec City in Canada, 18. So here we are, we have social media going, find out what we're doing. There's our webpage, and we have a mailing list, and about 2,000 individuals are on the mailing list for our newsletter. If you would like the newsletter, then please sign up on the website or send an email to us, and we will make sure you find out what's been happening with IUCAN. So please, Moldova and other countries that may not be involved, join a society, form a little group under a bigger society. You only need 10, 20 people to do that, and come and join the International Union of Film Applied Biophysics community. Thank you.
Thank you very much. This concludes the first part of the preliminary session. So let's go for the coffee break, and 11:30 we'll have to. Okay? With a small.
So good morning, and it's about time to start up our session. And the next speaker, Rainer Adelung, coming from Kiel in Germany, and will talk us about air materials and frameworks from global side, for biomedical sensing and energy researching.
Yeah, thank you very much. So, I'm very happy to be back in Moldova and continue, I would say now, this series of talks which ranged. I noticed that I was at the first conference here in 2011 already, from, I would say, basic research all the way to application. So I can also present now some news. So we funded a new company, Aeromaterials, last month, so which is now making use of these materials, which we thought first were purely kind of basic research. So the talk will contain basically 3 points. Our weird materials, so this tetrapodal zinc oxide, will be introduced, and already that they have some medical use because we could employ them as antiviral agent. Then shifting to aeromaterials, which are on this tetrapodal zinc oxide created, and then finally some applications, lately actuators, which are very energy efficient, and tissue templates, but it's also possible to make loudspeakers without any electromagnetics, and filters, sensors, diffusers, and so on. So, where does it all start? It all starts for me in Kiel. So this is an aerial view. If you were not there, it's beautiful. It's the Baltic Sea. If you would like to visit us, we are very much in the north of Germany, close to Denmark. For orientation, we are here now. So it's not too far away. So with 1-2 airplanes in good range. Okay, so what's with this zinc oxide, this tetrapodal zinc oxide? We do it in a very simple synthesis. So when I started my group, I didn't have too much money, and my title was functional nanomaterials. So therefore I had to show up with nanomaterials. Zinc oxide is very prone to form nanostructures because it has, in the C direction of the versite structure, a dominant growth direction, so it grows up very fast. And we created a simple oven synthesis, you see here, where we basically burn zinc powder together with a sacrificial polymer. And the polymer has just the job to be burned, so to catch the oxygen away as long as it's all consumed. And then the zinc oxide at once will be oxidized. So this is a trick with which you can even do nickel magnetic materials and so on to keep them away from oxidation. So afterwards it looks like it's snowed into the oven. So you see here a crucible. Typically this is showing a gradient where you have very large crystals, very small crystals, and you can tune all that towards, we call it flame transport synthesis, towards certain shapes. And what we would like to have is this tetrapodal shape. we can see that you can make it also on larger scale, and that's the idea of the company we started longer ago, the Firestone, which is doing this antiviral agent. So they have to do the synthesis more professional. So as you can see here, so they have a medical clean room. It all has to be GMP certified under strict quality control. And you see they're doing large plates of this tetrapodal zinc oxide. So how does it look now microscopically? Well, you can see this here from this image. So you see here the scale bar 20 micrometers, so a hair would approximately have such a diameter. So you see there's a lot of open volume between these tetrapods. So there's a huge free volume, and these are microscopic arms in between. So yeah, and the tetrapods were somehow very popular. I never get rid of that research. So over the years, we had many different work on tetrapods, and I wanted to kind of stop that, but it was not possible somehow because the group is coming with more and more ideas and applications for that. And even at this beautiful conference, we had a nice opening yesterday. You might have noticed that somehow the tetrapods, if you take a careful look at the background, sneaked in here somehow. The reason is maybe mathematically. So if you see the tetrapod is a very nice structure. So if you have 4 vectors, it's the most isotropic arrangement. So you see here 4 arms here, and they have all an angle of 109.47 degrees. So it's most widespread, and it serves therefore as minimal topology unit for the connectivity, and so therefore it is very good for stress distribution. So if you have one arm, the other 3 legs kind of support that, and so you have an ideal motive for also structure. So if you're interested in tetrapodal materials, this is now a bit aged, 2018. There's a review from Yogen Vakuma Mishra. He was in my group doing his habilitation, and his main work was kind of managing the synthesis and bringing that up. He's now professor in Denmark and still very active in tetrapod research. Okay, so what is this functions you can have from these tetrapods? So first coming to a biomedical application. So therefore I have to show you what happens if you're infected by a herpes virus. The herpes virus is this little thing we are all aware because of coronavirus. We are all trained and know how viruses work in principle. They have nasty tricks to get inside of the cell. They have some glucoprotein groups at the outside, and with this they're kind of going inside the cell, and then they're convincing the cell core to be reproduced, and afterwards they're also. So what was the idea? You might know, for example, if you have lip herpes, it doesn't look that pleasant and is quite, not very nice. So the simple idea was basically that, like you find in a pub, for example, in order to get rid of the flies. So the zinc oxide has a crystalline surface, and this surface can have defects, and we found earlier that it can bind to glucoprotein groups. So if you have a crystalline surface, then you have here an oxygen vacancy, and the trick is basically, this is out of scale, that you grab the virus by these glucoprotein groups and that you immobilize that. So that was a very simplistic view. I'm a simplistic guy from physics, so not too deep into biochemistry. However, it turned out that the virus is really small compared to the tetrapods, so a virus would be approximately the size of the laser pointer here, and we could nicely see in fluorescent images and in vitro images that you can immobilize them. And then there was some years passing by, so this was I think 2009, 2010, and then we were working with people in America, especially Deepak Shukla later on in Chicago, and he had a cohort of mice which are suffering from the herpes virus. And usually, if you expose herpes virus to these mice, they die already after 14 days, so the population goes really down. So then the idea was, maybe we can treat these mice a little bit with the tetrapodal zinc oxide and the cream, and therefore we will pull away a little bit viruses. Let's see if we see an effect. And it was totally dramatic and strange, because all treated mice, they survived. So this was done 3 times with a cohort of 20 mice, and it was totally fascinating and difficult to understand. And then Deepak Shukla, he found another mechanism which is responsible, and we could, in a petri dish, simulate that. Again, he did all the genetics and so on, what you need in order to understand it. But the story is basically this. Here you see a fluorescent marked virus sitting on the tetrapod being immobilized. And these are antigen presenting cells. So they are sneaking out if they find an object like a tetrapod and going all the way over the surface and eat up everything they find on this tetrapod. And they incorporate this herpes virus and then start the immune cascade by the CD4, CD8 signal pathway. Here, people who know this much better than me, I've learned it. Basically, the killer cells are then going on their target, and we could see that all these mice were immunized because of that. And that was very nice and beautiful. So first application for this tetrapodal zinc oxide was done. There's many publications, but I would say this one with Deepak Shukla is the kind of key publication if you're interested in that. Where he very carefully studied this. So then the company, which was already started, focused totally on this tetrapodal zinc oxide and on this fabrication, and then we could bring it out to the market, so that we have now a product which you in Germany at least can buy via Amazon even, where it says indeed tetrapodal zinc oxide here is in the ingredients. And that could be rapidly translated into the market, because zinc oxide is basically in every second cream, but not in this tetrapodal shape, but in a very small shape, where the only purpose is that the nanoparticles from zinc oxide should be dissolved. And this is here different. So here it's a kind of like solid state pharmaceutical effect, because instead of utilizing the atoms or ions to have an effect, we're utilizing here the surface and the surface binding. So somehow you could say, from perspective of a solid state physicist, the defects on the surface, so the surface doping sites, are doing here some biomedical action. So this brings us also at this conference in between nanotechnology and biomedical engineering somehow. Okay, you can do further stuff which we recently studied, so maybe I skip this basically, but one effect is that the antibacterial activity of this tetrapodal zinc oxide is different from the normal zinc oxide, which is just flooding the bacteria with zinc, and so therefore they have problems to handle it. Here it is that you have a different antibacterial effect, and a wound dressing was created where these tetrapods were combined with hydrogel and 3D printed in certain wound patches. Moreover, you could load those tetrapods if you change these surface binding sites with vascular endothelial growth factor. So for those, it was very useful to release those, which could be done then with light, in order, if you have wounds which are difficult to heal, have some additional blood vessel growth. So this was done with my PhD student Leonid Siebert, who's now doing his habilitation, and he went to the Harvard Medical School for that, and in this visit he could establish that. And here is this antibacterial effect, which is then different from the one in particular zinc oxide. So all I want to say is it might be useful to take a new look at these materials like zinc oxide and other materials, and examine them under the aspect of, I would say, solid-state pharmacy. Because it would be, I think, a strange coincidence if it would be the only one which would have an effect like this. We examined it a little bit with tin dioxide, for example, which is biocompatible as well, and we saw there that virus binding is possible as well. And of course, one should adopt then for certain viruses and antibacterial effects to tune the material in the proper value. Yeah, so this is later stuff. Yeah, as time is short, I would like to switch gears and go now to another feature you can do. You can take this powder and you can resinter it in all kinds of shapes. And this is shown here. And it's even possible to 3D print these tetrapodal stuff. This is with Hermann Seitz at the University of Rostock, in order to have, for example, gyroid shapes, in order to have a hierarchical porosity, which is important for biomedical applications, but as well for engineering applications. And after sintering, you've seen the structure again. You have a lot of free space between the individual tetrapods, but everything is very well engineered, connected. So it's a highly porous material. It has already porosities over 90%. What we discovered then, longer time ago, and I think in the second conference here, I was presenting that in 2013, that was a joint work with Hamburg, with Karl Schulte. He's expert for carbon CVD, and so therefore the tetrapods were taken, and with the help of a broken hydrogen valve, which we found out later, it was possible to deposit on the tetrapods a very thin carbon film, and at the same time, with the help of the broken hydrogen valve, we could remove then the zinc oxide underneath. And so, therefore, you obtain this same object like we had it before, this sintered zinc oxide, but now as a hollow framework, which is consisting basically out of no material. You see it's already in the scanning electron microscope transparent. So you have a material which is extremely lightweight. We could go down those days to 0.18 milligram per cubic centimeter. So basically lighter than the air in which you put it. And that means you have 99.991% porosity. So one variant you saw maybe yesterday in Jon Tiginianu's talk. We did it later on together with the gallium nitride, and therefore I have to also make a remark. He said it's over 90%, it's even over 99% in all cases porosity. So very lightweight, very open porous, but still very black, because if the light goes in, it gets scattered and then eventually absorbed and never comes back. Still, it's a robust stuff. You see an electrostatic experiment, which I like to show you. This is real time, this is no high speed. So you're attracting here the material with a charged stick. It's picking up the charge, and so therefore being expelled, because the material is conductive as it's carbon, and it's kind of bouncing on the table, distributing the charge on the table till it gets attracted again. Here you see it in slow motion. Wonderful physics, and it shows that with acceleration and so on, you cannot damage the material. It has also aviation and space certificate now, because we could apply it there. We had a project, for example, with Lufthansa Technik in the framework of the graphene flagship to build an air filter for an Airbus 320 from them. And there's another speciality which we found out with Nicola Pugno. He made a model of that, and then he found out that these hollow tetrapods, that they can be bended and go up, and that therefore the material can be super elastic. However, if the material is sticky like with carbon, then it's viscoelastic. Okay, so another variant which might be interesting here for biomedical community is that you can also deposit hydrogels on top and etch away the zinc oxide material. And in this way, you have a very nice template for cell growth, because you have a lot of free space in between here, and still you have channels which are all interconnected in the middle, with which you can supply the cells. And together with Robert David in Rostock, induced pluripotent stem cells were converted into these heart cells, which start then automatically beating. So this is inside now, this material in a 3D configuration, and Robert was saying that he never saw such a kind of strong beating and long-lasting beating. So maybe this is due to these channels. If you're interested, there's already a preprint on Chem Archive out of that. And how the hell getting here this aerogel and this thin film on top, that is this mystic ICVD, which was discussed yesterday already a little bit. And this ICVD is done by my colleague Franz Vaupel, but originally it was invented at the MIT with Karen Gleason. It's initiated chemical vapor deposition, and this is a very interesting technique to make polymer thin films. So, it's basically like a free radical polymerization where you have an initiator and some building blocks out of monomers, out of which you will build then your polymer, but it's just done in the ICVD thing. So it's a free radical polymerization, and the initiator will be breaking down in the vacuum chamber on a hot filament, and then very controlled, you start your polymerization like in a beaker. And they did 50 different polymers, and I think Franz Vaupel and Stefan Schröder, they did now 100 more, so you have a big variety about that. And I would emphasize that you saw that we are working with the University of Rostock quite a bit, so we teamed up. We are both poor universities for German measure in the north, and we all go interdisciplinary, and we found that we have a combinatorial explosion if we bring our stuff together, like shown here with this beating heart stuff. Okay, here's another application of this ICVD, very briefly. So if you have your conductive material, in this case not graphene, but in this case P.PSS as a polymer, you can, for example, deposit a Teflon PTFE around that, and therefore you can have your samples, for example, which are flexible shaped, so you can compress them and release them, and then you see if you coat them with Teflon, then you have only marginal change in the resistance during these cycles, and so therefore it can be, for example, one of the very stable systems. So as one application of the ICVD. So with CVD on these tetrapolar networks, you can do a lot of stuff, no matter if ICVD, ALD, or we could even do boron nitride. And now, I would like to use the last 5 minutes for the engineering applications we recently did with the new companies. Therefore, I have to introduce one formula, which is pretty simple. So, if you want to heat up things, you have to push heat energy into your system. And then you get an increase in temperature. But there's a proportionality constant, which is the heat capacity. And that's the specific heat capacity times mass. And if you change this equation so that you see what's the temperature increase, for example, from resistive heating by going with a certain voltage and current into your system for a certain time, then you see that you find the mass in the denominator. And that's very good because basically there's no mass, so meaning that you're dividing by a very small number, so you get a high number here, a large temperature increase with low power. In a millisecond, it's possible to heat the material, if it's from graphene, all the way to 400 degrees. So actually, you have heating rates of 500,000 degrees per second. So you cannot heat it that long up because it would evaporate. But we called it electrically powered repeatable air explosions. I said even reproducible air explosion, knowing that the referee will kill me for that. Okay, he didn't like it, so therefore the idea was to call this E-Pray, how we called it, repeatable. But I think it's even reversible because you're just heating air, so it goes all the way back to its initial state, and entropy is not larger at the end than before. We can discuss maybe in the coffee break if you're interested. What can you do with this? Well, here you see a cylinder, and this contains the aero material and nitrogen, and it's heated, and you can see that this piston moves up. Why is the piston moving up? Well, you have this open porous material, so you heat it dramatically fast. The air inside is heated dramatically fast as well, so the air shoots out and can move this piston. And yeah, so you see here, even if you put 2 kilograms on it, you have quite a good actuator. So for the view graph, if you see strain versus gravimetric power density, it's quite okay. Everything which is in the upper right corner is always good. So therefore, we had to go here with the gravimetric power density. Okay, should stop joking, but at least it's quite good, as you can see here, so you can utilize for something. The company which was founded will now provide this aero material in a contacted manner, and now you can use it, for example, to power pneumatics locally. So you do not need any kind of pump and tubings and oil filters and switches, but you can directly use a pneumatic piston and switch it on. This was from the Hanover Fair, where we presented it still as a working group. So here you see 2 pneumatics and waving the flag of Schleswig-Holstein, which is a local country in Germany where Kiel is located. So yeah, so this is thermopneumatic. And yeah, pneumatic is pretty inefficient, so even with our thermal process, we are fine, so we are even better than the normal pneumatics, so you save also energy, which is very important nowadays. Yeah, and one could say, yeah, it's without movable parts here, the piston is mounted to that, so you could say the material is the machine. I like that. Unfortunately, I found that somebody made the saying already 2005 for something different, but yeah, that's nothing we can do about it. Technology relevant is maybe the vacuum generator we can do. 2 weeks ago we filed the patent in order, 3 weeks ago, to present it at the Graphene Week. And the trick is that if you have your aero material inside of such a chamber, then you can heat it, of course, and the air goes out. But the air has to go back in, and it's soaked in. So if you combine it just with a suction cup, then you have a vacuum system with which you can do pick and place and stuff. And only the market for this small pick and place stuff is already over 100 million. So that's a good idea then to fund a company to try to sneak in and do that. And this is here now our prototype, where you can see here this little box up there, which just contains aero material between 2 circuit boards, and that's it. And yeah, with this we were, for example, sorting here these candies. Yeah. Indeed, 10% of the energy in the EU industry production goes to compressed air. So one part of that is in these pneumatic systems, so we hope that it's useful to utilize this for save some money. So, and it's sustainable by design, no rare earth materials, no movable parts, low energy consumption, and so on. Yeah, well, there are other actuators, but I'm afraid I'm running out of time slowly. One final thing is that, yeah, you can utilize them for filtration and utilize them also as loudspeakers. Because if you put a signal to this, you heat it up dramatically fast, but you cool it off relatively fast as well. So therefore you have enough for a signal. So then you have again a loudspeaker without movable parts, and it's very lightweight. So we are in discussion with Lufthansa Technik, for example, for aviation applications. Yeah, and this optical stuff, I think I skipped, I presented it here as well. So if you have the boron nitride variant of this aero material, you can scatter laser light that much that it loses its coherence, and so therefore, yeah, you have no speckle pattern, and yeah, you can utilize it. Together with quantum dots, you can even do up and down conversion. Anyway, and this was the final actuator we did with a gas-driven micro pump. Where we utilize the light all inside the material and some graphene to absorb it in order to pump here this water with this little pump without any electricity over the place. And Lena got the Bertha Benz Award last year for that, so I'm very proud as her doctor father to yeah, congratulate her once again in this. I would like to thank my group, which is overly active, as you see, doing all this work, and yeah, the people who are with me in the company and would like to bring now these aero materials based on the tetrapods forward for both for engineering and biomedical applications. So thank you very much for your attention.
Thank you so much, Rainer, for this so motivating talk. We'll have time for, yes, one question over there.
Thank you very much for extremely informative and interesting talk and presenting this great perspective direction of material engineering. The question is the following. Taking into account that their materials have extremely high surface, more than 99% porosity, it means that they will serve like extremely effective catalysts. Is it possible to extend other materials? Because for redox processes we need effective catalysts. Is it possible?
Yes. Yeah, we did, for example.
You could repeat the question.
Okay, sorry, it was it. Yeah, if you can employ it as catalyst as well, you have relatively large surface area and it's accessible. So the answer is yes. So you can deposit either catalytic particles on whatever substrate you want to do, but we could even do, for example, titania or ceria direct as catalyst material, as arrow material. So we are a material production group. So if you have ideas about how to measure that and so on, please contact us. We will bring it to a certain state, but if you have ideas and can quantify it, please contact me. Maybe later on during lunch or coffee breaks. We'll be happy to collaborate and send you the materials that we can try to work out something.
Thank you very much.
So, myself also at coffee break about the interaction with the biological membrane. We should move on, and I will hand it over to the other chair. Sorry.
Okay, thank you very much. So our next talk is on thin film radiation detectors for X-ray imaging, again from a Japanese attendee at this conference. Thank you.
Thank you very much. So I have an introduction. Thank you very much for me for IFMBE. I have to say very thank you for inviting me at this conference. I'm working from Shizuoka University, Japan. So today I will talk about thallium bromide thin film radiation detector for X-ray imaging detector. detectors. Okay, so I will talk about the introduction of thallium bromide. Maybe it is not familiar material for you. So, and then for making X-ray imaging devices, read out IC is very important. In our case, photon counting technique and direct charge conversion technique are put into the new read out IC. So, and another one is here. So, and I studied about cadmium telluride X-ray imager for so long time. So, I have compared thallium bromide and telluride. So, in this section, I use bulky times of thallium bromide and cadmium telluride. And finally, I will show the recent result of thin film thallium bromide. So, and I want to conclude. So, this is one of the results of the cadmium telluride, not thallium. Cadmium telluride based photon counting flat panel X-ray detector. We put this 1 cm by 1 cm modular like this one. So we make a 12 by 12 sensor devices. So maybe you can see the just mirror surface from here. So we developed this very flat tile using these devices. So this device is just prototype. So any collection of the image, X-ray image, there are also some fluctuations you can find. So we can get these kind of good contrast and low noise X-ray image from cadmium telluride. So usually two kinds of radiation detector is available we use. So this one is scintillator detector. In the world, widely used scintillator detector compared with the semiconductor detector. So in this case, X-ray photon comes into this material, scintillator materials. So we can get visible light. This is just converter from X-ray, the X-ray photon to visible light. So, and in that case, we can use a CMOS sensor like this, put on this CMOS sensor. So there also we can use the CMOS sensor. Now CMOS sensor, visible CMOS sensor is very good properties. Therefore, one of the possibility of the scintillator detector one of the reasons of the why scintillator detector is widely. So, after changing the visible light like this, light is scattered to any directions. There are low spatial resolution we can get, but it is easy to make a large size X-ray detector. So, by using the semiconductor like cadmium telluride, silicon bromide, and so on, we can put this kind of electric field. Then X-ray photon is coming into the semiconductor. So, and this X-ray photon made by these kind of polo electron pairs here. By using this electric field, almost of course little bit scattered, but almost moved to just the electron. However, we have to make read our IC. But we can get to the high spatial resolution. But it is difficult to okay, sodium bromide. So what is sodium bromide? So sodium bromide is a wide gap compound semiconductor material. The crystalline structure is a cubic CCA type. So and yellow-white crystal right side is melting point is also 460 centimeters. So just a table of physical property table of the many kind of semiconductors. So we use cadmium telluride so long time. So this is very good properties for high energy X-ray detection. So silicon bromide is almost the same, a little bit higher number of the atomic number and density. It is good for the high energy X-ray detection. So energy bandgap is so high, it is so low, it makes a low thermal noise and many kind of properties so good. So melting point is so I think it is easy to make a crystal of sodium bromide. Okay. So, if we want to make a semiconductor X-ray detector, so this table. So, however, this area, high number material is so good for the absorption of the X-ray. However, these materials so, have a very low band gap. It is difficult to use so, room temperature operation. Just now, so today it is so high dose sodium bromide. It has a so high density. Silicon is 2.33, so sodium bromide is 7. So sodium bromide is almost the same as high. So this is also absorption, attenuation absorption coefficient compared with so germanium, cadmium, telluride, and so on. This axis is also energy, so X-ray. So sodium bromide has so high attenuation coefficient. It means so high sensitivity devices. So this is a zone melting method to prepare. It's a preparation of the bulk sodium bromide crystals. This is a small heater here. So unmove this heater to this direction. Okay. This kind of just it is very fast, slowly and slowly and slowly. So in that case, so some material is not good for sodium bromide to move to this area. We can get so very high quality sodium bromide, single crystal sodium bromide. Okay, just experiments is not so difficult. Just graph tube is here. This is a heater pair. So this heater is moving so like there. No, some controller and some controller is being controlled here. This is also original material. Powder of sodium bromide is here. About 100 grams of sodium bromide. And then so we do this kind of so. This is the gross condition. The temperature is so low, so only 280. So, but the speed is slow and slow and slow. So we can get this kind of material, single crystal materials we can find here. So what the gross time is more than 20 hours and so. So what using this method? We can get so this kind of current voltage property. So we can compare with cadmium telluride data. So sodium bromide is only half of the small also that current. It is very good properties. So and we measure radio isotope spectrum. This axis channel means almost the same energy of the radio isotope. These are the curves. So we can find this kind of curve by using cadmium telluride. So in sodium bromide, so low energy is not so good for this material. So we can get this kind of equals 137. So little bit form is a little bit different, but also sensitivity is so high in both cases. Okay. This is also sensitivity number of the count. It is only in this high energy peak. So sodium bromide is very high compared with cadmium telluride. Cadmium telluride is not so much, but sodium bromide is more high sensitivity. we calculate the mobility lifetime product by using the hectoequation. So in the cadmium telluride case, it is so high. So it is so stable. So by changing the additional voltage. So in the saline bromide, so we can find the relation. So by using this data, so by using this equation, we can find this kind of metal products. So cadmium telluride is 3.5 by 10 to the minus 3. So thallium bromide is 2.03. It is almost the same. So by measurement, also with our developed devices. So next is we developed readout IC. So usually, X-ray photons is come into the detector. Detector make electron pair. It means of charge. So by using this kind of ADC and compressor, so we can get the spectrum value. The intensity is number of count per time, unit time. So but the X-ray energy, photon energy is count by the charge to this number of charge. Therefore, so very high speed of processing is needed. So this kind of circuit. But also our circuit is not so interesting. So but the generated carrier is moved to the integrator. Integrator is considered as a very large capacitor. So with current voltage converter. So by using the very small charge injection unit, injection circuit here, so we can remove the charge here so by probe like this. By using this method, we don't need any analog circuit here. So digital value of the number of X-ray to the change to the number of charge. It is difficult, maybe 100 or 200 one lowest unit. So, but we can get to the digital information by using this LS. So this is also photon of the prototype chip. This kind of small, but also it has some very interesting property here. So, but also in the cadmium telluride case, we need this kind of electrode, patterned electrode is needed. In this case, so we use platinum. So by using platinum pattern electrode by using the photon lithography method. So on some main now so we'll be using the silver, so but not so easy. Compared with the thallium bromide, we prepare the common electrode like here. Here is the thallium bromide. So but we don't use any pixelized pixels here. So and then so we put the silver band right here. So and just put on the thallium bromide, thallium bromide that put on the side. This is some pictures. This is the thallium bromide. This is that's brown. So it is nice. So we make this kind of material by using the silver paste. So we can get this kind of structures. And in that case, it is first time of the thallium bromide. Therefore, some spread here. So what we can get this kind of thing. So and we can connect with the silent bromide to so Adelung IC. So this is the example of the imaging. So this one is the background. Background is no X-ray. We use the photon counting technique, so we can get the perfect zero level on background. And X-ray only. So it is not so flat. So but we can find this kind of images. This one, this black line is scratched from crystals. So we put the gold wire 0.5 millimeter pi is millimeter pi is put on the detectors. We can find this black line from absorption of the gold wire. So we put these materials put on the samples that we cannot find with this character, Japanese character E, and without E, no changes. But so by using the cadmium telluride, thallium bromide, thallium bromide can detect high energy, so it is difficult to make contrast with the copper and so on. So in this case, we put on this material just slowly. So we can find this kind of images. Maybe I has past data also, thallium bromide parts. For reference, cadmium telluride image almost the same. So what we use here, so we can get this kind of clear images. So now so more clear. So this is the difference of the material. So, and we measure some modification transfer function, usually so we call it MTF by for thallium bromide imaging. We can this kind of here is so some metal here. So we can find the edge of the data here. It's current function that means like this summation. So we can find this kind of curve. Therefore, so we can measure so MDA values. So, however, maybe you cannot understand the detail of this view graph shows the MDA of the thallium bromide and cadmium telluride and CT devices. So almost same data get from these detectors, so we can say that it is good data. So however, so this kind of the connector is in the cadmium telluride kind case, so it is needed device because so cadmium telluride backside of the cadmium telluride has sterile electrode. So we can make this kind of structure. However, so thallium bromide is so we can evaporate low temperature condition. Therefore, so we want to make this kind of substrate by using this vacuum evaporation system. But crystal is easy to control the temperature. So we can get the single crystal of the high-end application. So, but this material it is very soft and has low melting point. It means that making a post-processing is very difficult. So by using vacuum evaporation, some utilities are available. So, but if we cannot use the post-processing this is a result of getting thallium bromide films. We can hide this kind of multi-peak by using XRD. So maybe polycrystalline here. It is same properties. You find things so with kind of surface. but so we can find this kind of columnar structure from these devices. So, and by changing the substrate temperature also vacuum deposition, so we can different grain size by changing the substrate temperature. By using this more, we want to use more high temperature, but the melting point very low. It is difficult to make room, so they want high temperature. Therefore, so we decided to use annealing process after deposition, after vacuum deposition. Substrate temperature is room temperature 70 degrees or 1 degree. This is a vacuum deposition. But annealing condition, we use more high temperature, so 210 centigrade using this. So this is deposition temperature. Substrate temperature, so deposition condition. So we use 210 centigrade annealing. So by using this annealing, so before and after this, we can change this kind of devices. By using the annealing process, we can get more large size of the grain from this material. So, however, so before annealing, so we can find very narrow grains here. So, however, so by using the annealing process, we can get this kind of wide grain size from. Okay, so this is the final result. So this is amazing alpha energy spectrum. So in that case, non-annealed and substrate temperature is maybe this red line is no radioisotope, but just noise of the spectrum. So, by using the amazing source, so we can find some data here. But
and we shall start our session number SE2. Short information. We shall finish our work until 15:30. That's why the reporters shall use 12 minutes for talk, 3 minutes for short discussion. That's why to be in time. Who has the extended version? For example, my version of report is 30 minutes. It will be during the plenary talk next day. But today it will Professor Lupan, me and some other colleagues, today short version, presentation of the main results, and the extended version who has plenary talk will be another day. Now let me introduce the first speaker, Maria Devalet, the report Advanced Biomaterials in Tissue Engineering, a critical review of nanocomposites based on bacterial cellular. Please, is reporter here? If not, then I have 12 minutes more, that's why I can present extended version. Okay, the second talk is mine, please, the second talk. Superconducting components for brain-inspired neural networks, spintronic innovation for neuromorphic computing. Please, the second talk. So that's why I will present within 25 minutes, 5 minutes for discussion. That's why we use our time. Okay, I would like to present results of tight collaboration in the frame of two European projects, SpinTech project, where we were coordinator, our institute, and partners from University of Twente, Netherlands, and University of Stockholm, University of Sweden. And Erasmus project, within 3 years we have tight collaboration follow up after SpinTech project and our national project. That's why it is interdisciplinary activity. Next slide, please. What I'm going to talk about. First, motivation, why it was necessary, not only interesting, but necessary to develop artificial neural networks. Then examples of already existing artificial neural networks. Then we will explain the necessity to meet the superconducting solutions for next generation of artificial neural network. New ideas, technologies for realization of these ideas, and then the two main base elements of neural network, artificial neuron and connecting element artificial synapse, and some conclusions and ideas for further development of this area. First of all, I would like to remind you, here the majority of people deal with microelectronics and know this famous empirical Moore law in development of microprocessors. Exponential growth of the possibility of computers due to reduction of size of single elements from 10 micrometers in 1971, when the first chips appeared in the market, up to 5 nanometers. It is already commercial technology which allows to prepare transistor on the 5 nanometer size. And further it is possible, but it is impossible. Why? Nowadays Moore law is exhausted and now it is plateau. Why? Why? Not only size we can reduce, but energy consumption and heating. My notebook produce like fan for hair heating, 90 watts. If we prepare even less size, then just can melt processor. That's why, the next slide please. All 4 generations of modern computers, including these multi-core computers, based on von Neumann architecture, where he produced to resolve the nuclear problem, it was necessary to provide huge amount of calculations. That's why a mechanical calculator could not do it, and he proposed the first computer, ENIAC, electronic numerical integrator and computer, based on vacuum tubes. It is original Great Britain production of vacuum tubes, and approximately 18,000 such vacuum tubes, 30 tons this ENIAC, but it with von Neumann architecture, it was so successful that 15 years, here on the photo you can see behind von Neumann, the first von Neumann architecture generation computer, ENIAC. The next slide. The second generation was based on transistor. Nobel Prize winner John Bardeen invented transistor based on 56,000 germanium transistor, was prepared in 1955. Filico 2000. Filico, Philadelphia battery company, it is IBM. Later it was renamed IBM. That's why the first IBM computer was based on germanium transistor. only 1 ton. Yes, only the notebook only 1 ton. The third generation of computer, next slide, was built in 1970 as we entered Technical University. First chip based on approximately 1,000 transistors. And the first IBM computer was UNIVAC IBM System 370. Original photo from museum. It is also based on von Neumann architecture. Next slide, please. And the last generation of my notebook, yours, is based on the cores, multi-cores, 3, 5, or 7 cores, but all of them consist of huge amount of semiconductor transistors. And the next slide, once again, one more. And here you can see the problem. In Sweden, where we had our FITEC project, there is one from 700 database center of famous Facebook data center. They have all over the world 700 such data center. Only single one data center based on multi-core transistors, multi-core processors, needs 9% electricity of a very good industrial developed country, Sweden. 7%, 120 megawatt energy. That's why the further increase of number of transistors, it is just impossible. You can see we just are heating environment. That's why to resolve the problem of energy consumption, need another architecture. Next slide, please. That's why fifth generation of computer was built using the natural computer, which all of us has natural computer, which needs maximum consumption only 20 watts. Instead of 120 megawatts, only 20 watts. Usual people needs much less. Some people don't use brain at all, but
still 20 watts.
But students who are preparing to examination, or women in store, this is huge work to scan all things on the shelves, to compare price, quality, size, design. It's a huge work. That's why during such work, brain energy consumption is maximum. Right? That's why it was hint from nature to change the architecture. Next slide, please. And our brain, natural artificial network, consists of two main elements: non-linear switch like transistor neuron, and linear element connecting neurons, which shall have the linear characteristic to change the connection between neurons from 0 up to 1, synapse. That's why neurons and synapse shall be connected in multi-layers, so-called perceptron or artificial neural network, which can gain information, treat it, and to give the decision. Next slide, please. First attempt, it was done many years ago, in 1968. You can remember that it was a postal envelope which stylized numbers, which we shall write the index, postal index. And it was for recognition by such system. The first commercial artificial neural network based processor was done the same IBM company, and IBM Trumor in 2014, the first successful processor, which consisted from 1 milliard of artificial neuron based on usual CMOS metal oxide transistors. Something like 30 transistors created one neuron. And linearization of nonlinear characteristic of transition, it is a difficult problem. That's for linearization of characteristic, one needs something like 35, 40 transistors to make artificial synapse. The next, please. And they constructed the processor. Here is 16 near processor. Each of them consists of 5 million transistors, and each simulates 1 million neurons and 250 million synapses. And it was enmarked 10 years ago. The next one. Here one can see the image of some successful commercialized non-phonemic processors. This is IBM, which I told already, and another North Pole. And the next slide, please. And this computer can very rapid. First of all, the most difficult problem is recognition of images. 90% information we gain from this device. And this is the most complex work for artificial neural network to gain information and to treat and to recognize objects. And that's why this task was resolved, and for traffic or you know nowadays without driver taxi or LKW, it is already nowadays no wonder that in metro train goes without lokfører driver. That's why it is good. This is the construction of such processor with elements. Everything is good, but not good enough. The next slide, please. Database center like Facebook, which I showed previously, which used 120 megawatts, based on this artificial neural network. For example, database center BERT, energy consumption 1000 kilowatt. It is 100 times less than usual database center, but in comparison with our brain, it is much, much more, and it is not good enough. Good, but better, less energy consumption, 100 times less, but a big reserve for further improvement. Next slide, please. That's why slowly I follow you to decision which you can make by yourself. The next step which shall be done to use superconducting based elements instead of semiconductor. Semiconductor elements, single transistor energy consumption very small, some femtowatts, but if you have 10 milliard of transistor, total will be 1000 megawatts. That's why if we use instead of semiconducting transistor with dissipation of energy, superconducting element, then the dissipation will be zero, not small, not less, but exactly zero. And such solution was naturally proposed many years ago. Just technology, you see that development of technology allows realization ideas. And based on the same base element, Johnson junction, superconducting Johnson junction, one can construct two different types of supercomputers. One, this is the analog of our brain, artificial neural network, consisting of neurons and synapses connected. Another one, more complex, superconducting quantum computer. Quantum computer, it is intrinsic probabilistic, and it is the aim of this computer to resolve some special problems, because the 99% of problems shall be resolved with computer for Neumann or artificial neural network. And the difference is in temperature level. The artificial neural network shall work at temperature when the elements are superconducting. That's why if we have, for example, niobium, helium temperature, it will be quite enough for operating. That's why for Kelvin, which our students yesterday made laboratory in our laboratory work, we demonstrated how easy to obtain helium temperature, commercial like refrigerator. It is a simple and cheap element for obtaining helium temperatures. But for quantum computer, there appears the quantum mechanical problem, so-called decoherence. Decoherence can be resolved only when we reduce temperature up to millikelvin. That's why the lower temperature, the longer coherence time when a computer does work. That's why for quantum computer, you see 15 millikelvin, this is usual temperature. It is very expensive, complex. It is a special direction of physics. I have course of low temperature physics. I explain to students how difficult each stage of temperature. It is one order of price in devices. That's why that is possible, but we will not speak about it. It is for special cases. We will speak about artificial neural network. Please, the next slide. To construct superconducting elements, it was proposed to use some phenomena. Hippie structures consisting of superconductor and ferromagnet. Superconductor and ferromagnet are antagonistic because superconductivity, for superconductivity one needs two electrons with opposite spins, which build the so-called copper pair, and copper pair is quasi-particle with spin 0. It is Bose particle, and Bose particle followed so-called Bose-Einstein condensation. In contrast to electrons, Bose particles they condensate in the single energy level, and they just cannot scatter on the difficulties. That's why no scattering, no energy dissipation, superconductivity. That's why in simple two words, what is superconductivity? But superconductivity, one needs correlated spin. Ferromagnetism shall make orientation spins in one direction. That's why usually it is antagonistic. But theoretical works proposed existence of so-called non-uniform superconductivity when superconductivity and ferromagneticism can coexist. The next slide, please. So-called Larkin-Ovchinnikov-Fulde-Ferrel state next slide, where Cooper pair can penetrate from superconductor and ferromagnet layer, propagate, reflect from the outside, and interfere, and one can observe like in Fabry-Perot, this ferromagnet layer for such hybrid structure serves like Fabry-Perot resonator, where if we can change the thickness of ferromagnet layer, we can obtain maximum or minimum pairing function. It means maximum or minimum critical temperature. Next slide, please. And in 1999, theoretician Takirov proposed to construct so-called superconducting spin valve consisting of two ferromagnet layer and superconducting layer in between. If we can imagine to change direction of magnetization of one of layer, then collinear magnetization, it will serve like a double thick ferromagnet layer, opposite only one layer. That's why changing magnetization of one layer, one can switch from maximum pairing function or maximum critical temperature to minimum, and to switch from superconducting to non-superconducting state of such element. And such element can serve like a superconducting spin switch. Idea is simple, but realization is not so simple. As usual, technology. And the next slide, please. In our laboratory, we deal with 30 years. We deal with thin film deposition. And here, this beautiful picture, one can see the structure of such sample, the standard wafers, silicon wafer substrate, silicon buffer layer. It is a technological trick to cover absorbed molecules of water, of oxygen, which can destroy superconductivity. That's why it is like a snow cover the surface, and it will be vacuum clean surface. And then on this vacuum clean surface, we deposit superconducting niobium film, then copper nickel, and another structure with cobalt. Cobalt has fixed magnetic orientation, and here one can see the change of critical temperature depending on thickness of ferromagnet layer. That's why one can really see the change from high critical temperature to disappearance of superconductivity and video appearance. That's why if we change the thickness of ferromagnet layer from here to here, then we can gain this Takirov spin valve. Next slide, please. Next. And next, please. If we just incline magnetization of one of the layers, ferromagnet layer, we can obtain so-called triplet superconductivity, and the critical temperature of such element can change from maximum to minimum. The next one. This is the construction of such triplet spin valve element, where we can normal metal just to separate two ferromagnets, weak ferromagnet, strong ferromagnet, and antiferromagnet layer to fix magnetization of one layer and to give possibility to change magnetization of soft magnetic layer. That's why if we change this weak magnetic field orientation of this layer, then we can generate triplet superconductivity and to change from superconducting to non-superconducting state. So the next slide. Here is, in this book, is described complete the all technological processes which were patented in our laboratory. And this is the German vacuum horse deposition, neutron deposition system labeled Heraus Z400. It is in majority laboratories or in industry. They up to now they use it. It is very robust, and one can with great stability to prepare samples using the system. So, and here we described all these ideas, technological processes, and the recipe how to prepare such superconducting elements. The next slide. This has been well designed. Two ferromanagnetic layers. One is with fixed magnetization with cobalt oxide antiferromagnetic layer, and soft layer where one can change magnetization. The next one. And here one can see how we can change the superconductivity by changing magnetization. The next, please. The next. And here one can see the switching characteristics of such element. The next, please. In this article, it is described the theory of such base element. This is Josephson contact consisting of two niobium layers and magnetic metamaterial consisting of thin niobium layers separated by cobalt magnetic layer. And here one can see how critical temperature change from parallel magnetization to antiparallel. That's why with the next, please, changing the magnetization, we can switch from superconducting to non-superconducting state. Here one can see the Josephson contact, which is cut using FIB, focusing ion beam. In Stockholm they have such device. I believe that in our clean room we also will have in the nearest future such device. This is only 50 nanometers, these holes. Here is the sketch of this device. Niobium layers, ferromagnet weak link, and characteristics, switching characteristics of such Josephson junction. And the next slide, please. And the artificial neuron based on one Josephson junction. Here is Josephson junction, and one can gain sigma characteristics like as this is the analog of sigma neuron. Using two next slide. Using two Josephson junctions. Using two Josephson junctions, one can create Gauss neuron or spike neuron. Our brain has spike neurons, spikes which activate neuron, and then they can be transmitted by synapse to the next neuron, and that's why this is analog of biological spike neuron. And characteristics depend on the strength of connection, inductive connection in this. Schematically, we can change the shape of this spike. The next one. That's why neuron is constructed from this Josephson junction. With one Josephson junction, sigma neuron, two Josephson junctions can give us spike neuron. More complex is synapse. The Josephson synapse shall serve, as I told, that linear connector between neurons. And theoreticians proposed to prepare the artificial magnetic structure, so-called metamaterial, consisting of the multi-layers from ferromanganese layer with different thickness. Curie temperature depends on thickness. For very thin ferromanganese layers, Curie temperature decreases. That's why if we have 1 nanometer ferromanganese, it will be weak ferromanganese, cobalt for example. If we have 5 nanometer layer, it will be stronger, Curie temperature will be higher. That's why preparing multi-layer structure with thin and with thick ferromanganese layer, we can, from the same material, convert to gain structure with weak and strong ferromanganese. And then we checked with experiments polarized neutron scattering. Polarized neutron scattering equipment allows us determine exactly spin configuration. That's why we can obtain from this experiment which direction of magnetization. Here one can see switch from collinear to non-collinear magnetization, only 20 ersted, very weak field, which we can generate just with a small current. For example, 10 or 20 milliamperes can generate such field and can operate this structure. The next, please. And it was predicted that changing the mutual magnetization, we can change kinetic inductivity. To make a synapse, we shall change the impedance of this element. Impedance, it consists of active part. In this case, active part is 0. It is superconductor. And inductivity is the another part of impedance. And superconductors, in contrast to normal metals, have additional inductance. Each conductor has geometrical inductivity, but superconductor has Cooper pairs, and kinetic energy of moving Cooper pairs give us the so-called kinetic inductivity. If superconductivity disappears, kinetic inductivity is 0. That's why below critical temperature in superconducting state, kinetic inductivity appears. It is small in comparison with geometric, but if we have very thin wire or very thin film, kinetic inductance will be comparable with the geometric. And this kinetic inductivity depends on the mutual magnetization of two layers. Here one can see dependence of kinetic inductivity of such construction dependent of parallel or anti-parallel magnetization of weak and strong ferromanganese layer. You can see that if we have thin enough superconductor, the thickness of superconductor layer in comparison with coherence length. For niobium, coherence length or size of Cooper pair is something like 8 or 10 nanometers. That's why if we have superconductor layer 10 nanometers, ions correspond to the thickness of superconductor layer equal to coherence length. That's why one can see that it is 30% change of kinetic inductance. That's why just changing magnetization of these layers, next slide please, one can tune the impedance of this functional structure. The next, please. The next. This is a real structure. This is the real structure which we deposited in our laboratory. Niobium 8 nanometer, cobalt 1.5 and 2.5, thick and thin. The next class. And we prepared split ring for measurement of kinetic inductance. Because how we can make measurements of impedance. Resonance frequency. If we can measure resonance frequency of such split ring, resonance frequency depends on sum of geometrical inductance and kinetic. Geometrical is constant, and changing the kinetic inductivity, we can change resonance frequency, and it will give us answer. Does change have we change of impedance or not? The next, please. This is the device for low temperature measurements, and the next. And here one can see the next one. The next here one can see the change of resonance frequency of such split ring by applying small magnetic field. That's why applying small magnetic field we can change kinetic inductivity of this element. The next, please. And this is real construction of artificial neural network based on such artificial neuron, this Josephson junction, and artificial synaptic connection between them. And this is the spikes response of such artificial neural network on the input signal. Input signal we can generate pulses like our eyes give pulses of voltage, and we simulate such pulses in inductive connected with artificial neuron. We generate such spikes and then penetrate in this neural network. The next, please. So that's why we have developed the technology which allows preparation of functional nanostructures which can serve like artificial neuron, and functional nanostructures which can serve like artificial synapse, and to connect them in the artificial brain-like neural network. And the next steps will be schematical. The engineer, for example, theoretic Alexander Penin, he is actively working simulating neural networks based on such element. That's why here one needs a lot of work for schematical development. Thank you very much for your attention. Any question? Any question?
В каких температурах работают квантовые компьютеры? Диапазон температур.
Значит, the question was which temperature one need for operating with such systems. For artificial neural network computer, one need very simple low temperature, low enough for superconductor transition. If you have base elements made from niobium, it will be quite enough helium temperature. That's why 4 Kelvin it will be enough. If you are developing quantum computer, now 4 countries, United States, Russia, Germany, and Germany in Karlsruhe, very developed. They constructed a 20 qubits computer. They need millikelvin, 10, 15 millikelvin. That's why it depends on the system which are developed. Artificial neural network. Helium temperature, it is simple, cheap. This, especially this system, need helium temperatures.
Pardon me. The neuromorphic chips, they are already in existence.
Yes.
I do not know the temperature they operate, but apparently it's reported in literature that
room temperature. They operate. They just need cooler just to prevent overheating. But as they are working on the transistors are working at room temperature.
So you know what's the material for this existing neuromorphic chips?
This is a smooth silicon metal oxide transistors.
Because it's so secret that they have not disclosed the architecture of neuromorphic chips. So,
Architecture is secret.
Right.
The technology is not secret.
right.
As one say, devil is in details. Everything is clear, but to repeat is very difficult.
But the chips are actually made in Taiwan. Those neuromorphic chips, they are made in Taiwan. So again, like I said, I'm looking into it, but I do not know the architecture and the material because it's all still a very high secret.
You see that I just give the very simple image. The problem is much more complex because here there are a lot of complex questions. For example, such chips they shall be done in 3 dimensions. That's why the technology is extremely complex developed. Right. Such complex only in Taiwan, in Silicon Valley, in Germany, in 2 places where it is developed. That's why theoretical it is simple, but in practical it is. Please, Professor Fomin, yes.
Thank you for your persistent presence to me to follow your excellent development in this field over decades.
Thank you.
Have you observed in your experiments the macroscopic tunneling through Josephson junctions? In the same style like Clark, Martinez, and Devoret.
You see that the tunneling of Cooper pairs, each student observed during Josephson experiments, but not each student understands what it is quantum tunneling. That's why it is the philosophical question. What is the difference to see and to look? To look can everybody, to see not everybody. Clark could see, and he constructed squid magnetometer. In Trent we worked with squid magnetometer which Clark constructed and as present gave to Trent University. Because Clark is a very clever experimentalist who 90% he spent to think and only 10% to do something with hands. That's why he belonged to the very rare part of population who can see, not only to look.
But to the credit of students, tunneling is not binary, it's quantum mechanical.
Quantum mechanical, yes.
So being complex and quantum mechanical, not being binary, obviously it's complex to understand for students. So I just want to give them credit for it because of quantum nature of it.
For example, Andreev reflection, no physical penetration, but reflected quasi-particle and looks like penetration of particle. Yeah. That's why it is difficult to imagine, but in quantum mechanics it is so that particle can exist, not penetrated there.
Sure.
Yes.
My question from Finland. I want to understand what do you think about the hypothesis that the nanotechnological scientists must be not only expert in the physics and so on, but also sometimes, from time to time, they should understand how its finding could influence, could impact to society, to smart city ecosystem, to smart city activity. And hypothesis is sometimes it could be. So, for example, in the conference of nanotechnology could be both the experts sections, but also, for example, places and event to understand how it could be benefit, what type of benefit could be to the society, to the future, to the different type of future. For example, what do you think about, this could be related to the education of students, yes, but to the education practice, training practice. What do you think? Is it to both of scientific, nanotechnological scientific activity would be interesting to the nanotechnological society? Because you have a very interest in nanotechnological society. And so scientist is a person who understands in the indicated and so on, but also in this culture there are different skills, different views about how it could be related to the life, to the development.
Thank you very much. It is a good philosophical question. You see,
practical.
No, it is not practical. It is not only for students, it is the whole society. This question was raised by Albert Einstein, who created so-called Pagos movement. Because he was not only a physicist, he was a philosopher, and he thought about the influence of great invention on the society, not only for students, not only for the education, but for society. And in Germany, I like Germany very much. I spent 12 years permanent, and nowadays we permanent collaborate with Germany. In Karlsruhe, in Forschung Center Karlsruhe, 30 years ago was created, Professor Adelung knows that in Karlsruhe does exist so-called Institut für technische Folgenabschätzung. Institut für technische Folgenabschätzung, auf English it will be Institute for Investigation of the Followings of the Technical Progress. It is approximately, it is not exactly a translation, but the meaning is the same. In this institute are working more than half philosophers.
53 minutes.
Huh? What?
53 minutes.
Exactly. You should a bit earlier. That's why the influence of the great invention, this is a very complex and important problem. That's why the whole institute, 30 persons are investigating the influence of nanoparticles. For example, nanoparticles, which Professor Adelung today explained, is very useful for medicine, but at the same time they can penetrate through skin and can kill person. That's why the investigation of the results of implementation of progress, it is not only for scientists, it is for philosophers. Thank you very much. Excuse me for a long discussion, but my talk was 30 minutes. The all was discussion. Talk
You are chairman, you should manage this.
So, thank you once again for your attention and very good philosophical questions, because this...
From Finland, this is foresight questions. Foresight questions. Methodology, it's not philosophical, it's a practical foresight, corporate foresight.
Institute for Technische Folgenabschätzung, they have,
Yes, well,
they have
not philosophical.
contracts for investigation. Yes, okay, thank you. Thank you very much. Now we shall follow to our program, and if the next speaker is here in the hall, please, Professor Manuel is here. Please, we will present how semiconductor terminology has been enriched by research of electrochemical pure porage and electrodeposition.
Thank you, dear Professor, very proper introduction. So the goal of this talk for today is to highlight some new terminology in the porosification of semiconductors by means of electrochemical etching. So electrochemical etching is the cost-effective and very accessible technology. And yeah, we started to develop this porous entitled compounds in Moldova. So the direction was initiated by Professor Tiginiano, and we started with the creating of National Center for Material Study and Testing at the Technical University of Moldova. So we have more than 2 decades working in this direction. So the next slide. I will be very quickly background and the evolution of the porous material. Some from porous silicon, because it was first material that was nanostructured, I mean it's ordering and so on, and to direction to the functional nanostructures. Also our contribution in this direction, and some application, ramification of the porous materials that we started and we developed in the Technical University of Moldova. The next one. Porous material, everybody knows nowadays it's a very important material because it can be filled with some other materials and play a role like some skeleton for another functional material. So the phenomenon was discovered many years ago, so unintentionally, it's not was planned, but this some side effect later was developed, and in 1990 it was obtained porous silicon. I mean by electrochemical etching in very controlled fashion. The next one. The interest in the porous materials was quickly increased after discovering of the photoluminescence in porous silicon, and was extended to another materials, I mean semiconductor compounds, because they have some advanced properties, I mean larger band gap, higher electron mobility, that give the possibility to use in another very important application in different domains. So the next one. Okay, so at the beginning porous materials were used like skeleton to fill them, like I mentioned, but porous semiconductor material give another possibility to use just the porous skeleton for, I mean, optoelectronics, to enhance the luminescence, or even to catch the light. And from the another side, it is possible to manage the conduction in this porous skeleton, just adjusting the pore wall's thickness, higher or thinner pores, and it is possible to stop electrical conductivity even in these materials. So adjusting the diameter of pores, thickness of the wall, it can be used in different materials. And of course, they can be filled with another materials and to create a hybrid semiconductor, for example, metal materials that I will speak a little bit further. The next slide. Like I said, more than 2 decades ago, we developed porous materials and introduced single crystals of nanopores. That means that in bulk materials, in controlled fashion, introduced pores, and like you see here, the even self-ordering without any photolithographic means it can be obtained, and this is due to the issue that in the material can be obtained 2 types of pores. Current line oriented and crystallographically oriented pores. I will explain in the next slide the difference between them. So current line oriented pores, they grow so straight, parallel with each other and cannot intersect. So after some time of etching in several seconds, even very high ordering can be obtained. From the other side, another type of pore, crystallographic oriented pores, they can intersect each other. So they create very interesting morphological 3-dimensional structures. And a lot of study was so done in this direction and compared in different semiconductor compounds from the group 3,5 and 2,6. The existence of these 2 types of pore give rise to the property to manage the direction of pore propagation. Like in this case, you can see some 2-dimensional propagation, so different degree of porosity in each layer, or even to switch from higher anodization voltage to lower anodization voltage to manage in 3-dimensional layer structures. And of course, it is possible in the same plane to have different degree of porosity. For example, 200 nanometers diameter and even 50 nanometers. So this kind of pores can give another response in optical properties, for example. Okay, so until now a lot of work was done, but gallium arsenide is very interesting material and it's well known, but also it's until now it was demonstrated that can be obtained current line-oriented pores. So several PhD theses was dedicated to this direction, but unfortunately it was not demonstrated. In this regards, we started to study how to manage these crystallographical pores and to obtain different shape of materials. So it was very interesting to use different orientation of the crystals, because from another side, the same crystals, for example, orientation 111, it have 2 sides, A and B. So from the side A, we have just intersecting pores, crystallographic intersecting pores, and give rise to the 3-dimensional porous structure that it was introduced the terminology Moldavite structure that was observed in Moldova. Another interesting issue that from another side, it is possible, after some optimization at lower applied potential, to obtain crystallographic, because the direction of pores, of growth pores, is the same like 111B. So it is possible to obtain to mimic some current line-oriented pores, but it's crystallographic because in cross-section it's triangular. After optimization of applied potential, it was possible to obtain in the same process nanowires with intersection of the pores that grow perpendicular to the surface and pores that can intersect. And as a result, it was obtained nanowires with modulated diameter, or even nanowires that are penetrated with pores. So it's a very interesting morphology, because we can change the conductivity through these nanowires that are perforated nanowires. The next one. The crystallographic orientation of the substrate it's very important in anodization, and it was demonstrated that we can obtain not only circular shape of the pore, but also square, triangular, nanowires, and changing the electron concentration of free electron in the material, we can change the density of these pores. That also is important for a different application. The next one. The time is not allow me to show you all the results, but we started to elaborate based on the porous material. So at the start we optimize the technological condition to obtain different shapes and to change the direction of propagation of pore. And after that we started to elaborate hybrid metal semiconductor structure based on these porous materials. So the workflow is very simple. So semiconductor crystals, the first step is electrochemical etching. It's formed porous membrane or porous layer on bulk substrate. And after that, in the second step is it started pulsed electrochemical deposition. So adjusting the parameters, it's important to have some pulse amplitude, the pulse duration between the pulse, and the pulse between the pulse. So it's possible to decorate with nanodots, nanowires to grow inside of the pores, or even to deposit nanotubes on the pore walls surface of the inside this skeleton. So some more information in the coatings journal that was published in 2022. Okay, the next one. So the one important technology elaborated and demonstrated is Hoping electrodeposition. When the deposition on the semiconductor surface it started, the dot are nucleated on the, randomly. Yeah? Randomly is nucleated. So the dot grow up to, in our case, it's 20 nanometer diameter. So it's a critical threshold because it's a Schottky barrier, because here is gold or platinum or indium phosphide, gallium phosphide. It's a platinum contact give us Schottky contact. Previous one. So when the dot reach up the diameter of 20 nanometer, so the mechanism is not convenient to grow up further. And the nucleation started on another side. So in this case, the nucleation jump from one side to another side, and as a result, just on monolayer of the dots with the diameter of 20 nanometers are covered the surface. So regardless the morphology. So it can be nanowires, porous structure, and so on. So every surface will be covered with one monolayer, of nanodots with 20 nanometer diameters. Okay, let's. So another important insight in this direction is integrated lens. So having these porous materials inside of the crystals, filling them with some materials, metals, it was demonstrated that we have some novel focusing element or even beam splitter. So depending on the distance between these pores, the thickness of the porous walls, and we demonstrated different focusing element based on some parameters. The next one, moreover, it is possible to change the shape of the metal nanotubes, just changing the shape of the pores in semiconductor materials. So previously we have optimized the technology. So having very good electron conductivity in porous skeleton, the metal nanotubes will follow the surface inside of this skeleton. So as a result, we can obtain different triangular nanowires. to manage the shape of the materials at nano level, it's very expensive and it's very difficult. But here, just by anodization, some optimization of electrical parameters, you can obtain nanotemplates with, for example, rectangular, square pores, triangular, circular, and to deposit inside the nanotubes. Okay, the next one. So as a conclusion, it's a very quickly. So this technology was recognized in the electrochemical society, and was a lot of citation. But the more important terms was introduction of current line-oriented course, current crystallographic course, semiconductor sieve. So when the porous layers are detached from the bulk materials, and the porous materials is open from the two sides. Okay, thank you very much, because we are lack of time. If you have some
questions, Professor Fomin.
Thank you. It was exhaustive presentation of long years. Some time ago, from your group, I think it was Mihai Inage, came to our institute in Dresden, and we investigated very interesting phenomenon which you have not mentioned. A fragment of this lattice of pores and the illumination, UV illumination, can generate bubbles. And in this way, it starts moving. So it's a kind of a rocket.
Yes,
still
micro submarine. That was introduced like micro submarine. Okay, that
micro submarine. What was the developments in that?
Yeah, the porous layer was nanotubes of titanium dioxide. Yes, so it's a very photocatalytic. Also can be decorated with some metals and to increase the catalytic activity under the light. Yeah, it's but it's very the long story, but it's very important development. Also, change the most important in this development is to changing the diameter from the larger one to the thinner one, because the bubble must
in one direction.
Yes, okay.
Yeah.
So thank you for mentioning this, because we not so many times, so I just miss it. Thank you.
Thank you. Other questions? If not, thank you.
In the coffee break, we can discuss.
I have some questions, but hybrid nanomaterials for biomedical sensors. So, please.
Hello to everybody. So this is a short version of our plenary from Friday, but it was included also in this section, so that's why I'm really happy to show you a short advertisement of our work and to see all of you here in Moldova. So mainly it will be a work done between several groups. Our group in Moldova, Professor Rob from Belgium, Professor Ameri from Kiel University, Rainer Adelung, professor here, and Franz Baufeld. So now we can see that trend and development of new types of materials, namely on hybrid materials, and to make different types of such sensors, which very close will be even implemented in our body and also on clothes and so on to monitor different gases. This is our field. That's why I will focus only on gas sensors based on these new types of materials which can detect different volatiles. Next, also I showed that it can be implemented in batteries. This is one. Batteries are also implemented in various medical applications and devices. And for these hybrid materials, it's very important to use different types. So we'll use organic and non-organic materials, combinations of this one to use their synergetic properties and to try to identify different types of volatiles from the brief and from to detect different diseases early as possible. So main idea for this work is on ICVT, like, yeah, this is new technology. Yesterday I showed you shortly also, but on plenary we'll discuss more about this new initiated CVT. CVT everybody knows and utilized for many years, but this initiated one uses, for example, monomer A, monomer B, and very easy hot filament in between, and it can be deposited at room temperature and on any types of these sensors, and different polymers can be combined and deposited on top to create these hybrid devices. And during anniversary of Professor Faufel group, he just suggested let's try to make some copolymers. So by mixing different types of polymers, PB3, D3, TTF3, and he just suggested, give idea. We went with Professor Adelung also, and we fast implemented and obtained such materials. It is not so easy, but it was done and confirmed by FTIR also and other techniques, these materials. Next, it was deposited on top of different types of oxides. For example, in this case, it was titanium oxide, and we used other types. And by annealing this, for these types of materials, it was done also theoretical calculations, explained what's happening inside, how this electronic fields affect affects their surroundings, so how gas can pass through these types of polymers. So we are trying to develop all this technology and publishing part by part in different journals. So here we have showing different polymers. For example, before, if it is as grown, it shows one type of molecules, but after annealing, it's transformed. So in this way, we are trying to prove, to explain what's happening when we are covering completely these oxides, creating hybrid sensors, and to explain detection of gases. So this is main responsible for he is a biomedical engineer, now PhD student, Mihai, and he is in Rainer Adelung group in Kiel now. Professor Adelung is here, he is in Kiel working. So we are trying to develop such types of hybrid biosensors to detect different types of gases, and also on scaffold in previous work, another joint project between Technical University of Moldova, Medical University of Moldova, and Kiel University. Next is a Horizon project. This is a joint doctoral school, big one, about 10 different universities working, and we have even joint PhD we'll have soon. So it consists in combining these hybrid zeolite materials, and we also use oxides and found a new types of devices. Part already is patented in Germany. Now Kiel University is trying to patent in US, yes, and develop further. So it's between 10 different universities. Main university is Kiel University, and also our students visited this nice laboratory in Kiel, and we tried to make a very good collaboration with Professor Adelung. You can see he is only one professor and a huge group of PhD students, and he is taking care of big power, big intelligence, and a lot of ideas every day. So and in collaboration with Professor Rob from KU Leuven, from Leuven in Belgium, which is main in this project, Horizon project. Also Professor Faufel, I showed, he just retired, and the new Professor Amiri is instead, and this nice group working on polymers is not so easy, like I explained yesterday shortly. It's a new technique, and only they can make these types of hybrid materials. I think in Europe nobody can.
A few maybe.
A few many, but about sensors only we did it, for sure. Yeah, acknowledgement to this Horizon project. This is just advertisement for our plenary talk on Friday. Thank you.
One short question. If
plenary.
We will write your questions and to rise in the plenary. Okay, thank you very much.
It's very thin.
Now, briefly to the next one.
It was calculated, it's less than nanometers. So it's tuned, this cage. Dimension.
The next talk, Елена Акимова is the next talk, applications of photosensitive composites for direct holographic recording. Please, Константин Лосманский. Константин,
Good afternoon, everyone. This is, let's say, second part of the STM presentation of Veronica Kuzak. So, my name is Konstantin Losmansky. I came from Institute of Applied Physics of State University of Moldavia. And today we'll talk about some applications and some properties of nanocomposites used for direct holographic recording. So, a short introduction. It's well known that azodized molecules has this property to photoisomerize, and this process is cyclically till the molecules reorient during this photoisomerizing reaction and reorient perpendicularly to the electric component of the light field. Also, during this process we have second moment. The size of molecule changes two times. We have for trans as a benzene molecular derivative we have 9 angstrom, and for cis we have 5.5. So this is first what we need to take account. Second one, the base, the polymer that we used for our study is polyepoxy propylcarbazole with solvent yellow 3 azodized, commercially available. So what we did, we just opened the epoxy group with primary amino and polymerized this system. And after we introduced gold nanoparticle. Why we introduced gold nanoparticles in polymer matrix? Because first we observed that first you all know that nanoparticles change the properties, physical properties of material. Today I will talk about optical properties of the nanocomposite. And the second one is for this movement from trans to cis, azobenzene molecules need space, a lot of space to reorient effectively and nanoparticles create this free space. This is another approach. Next slide, please. So the motivation is why we are doing this nanocomposites is for we obtain, using holographic methods, we obtain diffractive optical elements, and this is a microstructured or nowadays nanostructured nanopatterns that can change phase, that can change amplitude and direction of light, and also permit to create very complex and very interesting devices that it is impossible or is hard to get with traditional refractive or reflective optics. Then the second one is they are very small structures. In our case, for example, we have around 1 per 1 square millimeter. And Third, it must be important that these diffractive elements can be recorded, can be obtained using a real object, or we can use computer synthesized hologram to record using optical methods on the surface. So here I showed two types of vortex. This is an optical element used for optical scissors and tweezers. So in case of amplitude vortex, we need to know refractive index of the material, also to know the to calculate this height of it, to get expected optical element. In case of diffractive optical element, we just simulate, we just record or synthesize mathematically the grating and record on the surface with thickness of up to few microns. So the aim of this work, it was interesting to understand what happens in first diffraction order with material during the holographic recording, because we have not only the amplitude, and also was interesting what happens with polarimetric parameters such as azimuth and ellipticity for nanocomposite film doped with different concentrations of gold nanoparticles. And recording and to understand complex this behavior, the recording was carried out on 3 polarization states. But now I will talk about just one of recording beam, and also we take into account the polarization state of the probe beam. Next slide. So film preparation is already published, but we just use reflux method to obtain polyepoxy propyl carbazole solid yellow 3 polymer. After okay, we used commercially variable nanoparticles, but in our case, because we have toluene here, non-polar solvent, usually we're using Pierce Schifrin method to obtain these nanoparticles, but here we had used commercial because the distribution of size was very more or less the particles have the same size. Okay, and we mixed the solution in ultrasonic bath, and after we are using a little bit different method of film deposition is rod coating or bar coating. And, we can obtain from nanometers up to microns, just changing the height of this rod, and also for the same polymer. And also we can hit this on gray substrate to get different types of stress in the polymer. So we obtain, in our case, we obtain 6 samples with concentration from 0, means just ETC polymer, and with the step 0.2 micrograms per milliliter, obtain 5 samples with thickness shown in this slide. Next one, please. This is optical setup used for during holographic recording, and also for record polarimetric parameters. It is just a simple interferometer. We are using blue laser at 473 nanometers. Polarized beam splitter. Here we have 2 wave plates. We can obtain all kind of polarization, means linear and circular, and also in case of vortex, because we are doing this, also we can introduce transparent optical element in one of the beams and to get the hologram on the surface of the polymer. And also for probe beam, for probe laser, we changed a little bit from the setup. We are using bobination compensator to get precisely the polarization state, because this is most important for polarimeter. And this one is connected to computer to get in digital form this data. Next one. So both curves for diffraction efficiency and for polarimetry are divided in 3 parts. We have A, B, and C. A means when the blue laser is turned off, so we don't have any excitation. This is the initial state. B is during the holographic recording, and C is for relaxation. So blue laser is turned off, but we record what happens without excitation. So we name this relaxation zone C. Okay, next slide, please. So to understand how will our material will answer to blue and red laser was recorded obtained spectral dependence of absorption coefficient. You can see that for blue our material will be absorbed, and for red we don't expect any absorption, any interaction of material with this wavelength. Also, in this case, it's hard to say what changes we can see with the concentration, changes in concentration, because we have to plot to make a Taos plot, but in the future I will present this one. So, please next slide. So here we get 3 plots of diffraction efficiency recorded for polarization state of recording beam from plus minus 45 degree, and for 3 different polarization of the probe beam. What I want to show you, okay, it's hard to see here because in the case of diffraction efficiency we can show this segment C. When we stop the recording, we saw a difference, a relaxation, but it's not yet relaxation because diffraction efficiency can increase or decrease. It means that something happens in material after the exciting laser is turned off. Can I have the slide, please? So this is the briefly diffraction. I plotted maximum of diffraction efficiency for 3 polarization of recording beam versus concentration of nanoparticles in other polymer film. So the best result we get for vertical polarization of recording beam at concentration 0.08 microgram per milliliter. The question was why? Because this is interesting, not just simple behavior, but why? We're going to have next slide. No, come back. So, on this slide you can see polarimetric parameters, plotting of polarimetric parameters such as electricity and azimuth. And the first approach, you can see that nothing's clear here, but we found interesting moment that you can see here peak, here this one, and just few, in this case is around 10 to 20 seconds from the beginning of the recording. We get these peaks here, and in case of azimuth, these peaks appear. We think that these peaks can be attributed to the movement of the reorientation of molecule inside the volume of this thin film, but for electricity we found that this average from the beginning to the maximum can be attributed to absorption of the system of light with specific polarization. So, next slide. And here, why this happens? Because to answer to the previous question, because this polymer exhibits property to absorb some to absorb light with a specific polarization, and when this absorption is has higher values, in this case we get higher values of diffraction efficiency. As you can see, for example, for this case, we have electricity gets maximum value, also diffraction efficiency gets maximum value. So, in conclusion, here we showed in this work, we studied this nanocomposite based on polyepoxy propylcarbazole 7-PL-3, gold nanoparticle. Second one, we saw that this material showed these changes in thickness during the recording, and also it showed that we have modulation of refractive index. We get the high efficiency, diffraction efficiency for ±45 degrees, for concentration 0.8 micrograms per milliliter, and we reached 33.35%. And also, no, it's okay, I made a mistake. But polarimetric analysis showed that our material exhibits photoinduced difference, linear and elliptical one, and this one is in correlation with diffraction efficiency, and we think that this study offers new ways for developing advanced effective optical elements and different data storage system. Next slide is the acknowledgement worked by the project, and thank you for attention.
Thank you. One short question, because we have already time exhausted. You have a question? No. Okay, thank you once again. The last talk of this session, D and 3 nuclear isobutyrate oil complexes, synthesis, structure, and biological activity. Please.
Good evening, everyone. It's my great pleasure to present today recent research intituled D and 3 nuclear copper-2 isobutyrate complexes, synthesized structures, and the biological activity. Copper coordination compound attract attention to their redox reactivity, biological relevance, and interaction with the biomolecules, biomedical, agriculture, plant protection, fungicides, and bactericides. In this context, our study focuses on the development of new copper coordination compounds that have promising biological properties against resolving microbacterium vitis. We synthesized of 3 new compounds starting from copper-2 isobutyrate and various organic ligands by reflection in dimethyl sulfoxide or acetone under aerobic conditions. The solution were left to crystallizing slowly for weeks, yielding large well-defined crystals suitable for structures and the biological studies. A single crystal X-ray diffraction study of the compound is performing using of X-ray diffractometer. The crystal structures will solving using Shulk's program. Biological activity assays will conduct of the Public Institute, National Institute of Applied Research in Agriculture and Veterinary Medicine, and ER and gas spectra were recorded at the Center of the Physical Chemistry and Nanocomposition of the Institute of Chemistry. X-ray analysis showed coordination compound 1 and 2 are centrosymmetric binuclear compounds in which 4 isobutyrate ligands are connected to the copper atoms through 4 oxygen atoms, where square pyramidal coordination surrounding of the copper-2 atom is provided by the coordination in the apical position of the dimethyl sulfoxide 1 or 4-pyridine carboxyldigide N2. The molecular structures of the compound and the fragment of the structures excited along the B-axis are representing in figure 1. Compound 3 is trinuclear linear complexes. Here the central copper atoms is coordinated by 6 oxygen atoms from isobutyrate and triethanolamine ligands forming a square pyramidal geometry. The peripheral copper atoms also adopt square pyramidal environments with coordination involving both oxygen and nitrogen atom from the triethanolamine. The trinuclear units are further stabilized by hydrogen bonding network in the crystal leading to the one-dimensional chains. Hirschfeld face analysis. We perform Hirschfeld analysis for compound 1 and 2 to investigate intermolecular interaction. The analysis revealed what the predominant interaction HO and HC, highlighting the main forces stabilizing the crystal structures of the compound. And biological studies. Or antibacterial activity of compounds 1 and 3 against resorbing vitis was evaluated in vitro using the agar diffusion method. Experiments were performed with duplicate for reproducibility. The compound showed moderate activity compared to the tetracycline compound 2, production the large inhibitor zone. These analyses were performed to assess the potential of the synthesized compound and antibacterial agents. In conclusion, the investigation of copper-2 carboxylate base coordination compound is particularly relevant in the context of the promising antibacterial properties, particularly in terms of the ability to inhibit the growth of the resorbing Agrobacterium vitis. This investigation such the potential for further development of this compound and agents managing growth designs. Thank you.
Any questions, please, to the reporter? Clear. Thank you very much. We wish you great success. Thank you very much. Now, for the break, please.
Surprise.
Good afternoon, bună dimineața, bună ziua și добрый вечер. Welcome to the second part of our session for today on nanomaterials and nanotechnologies. Nanotechnologies and nanomaterials, sorry. Okay, as I told, this is a very exciting session, obviously, because it was longer than it should be. That's why probably many people did not manage to have the proper coffee break. That's why they will probably join us later. But since we are already 4 minutes late, let's start. I'm chairman. My name is Suri Kojokari. I'm chairman of this part of the session. And also, I will be the first presenter. Presentation. Yeah. Okay. Good. So, low power millimeter wave radiometer technology for Earth observation space missions. let's put to the session. Let me introduce you. Probably many of you knows what is a terahertz technology, and but probably not all of you know, because it's a multidisciplinary conference, and probably it wants to introduce you shortly in electromagnetic spectrum and particular terahertz region. So you see here, that's the whole electromagnetic spectrum. It's a natural resource like water and ocean. It's a lot of, but it's limited. For air and atmosphere, it's a lot of air, but it's limited. That's electromagnetic spectrum. It's a lot of resource. It's measured in hertz, but it's also limited. So it occupies first low frequencies occupied with radio waves, microwaves. Then we have infrared. So the visible light is everything we see. It's also electromagnetic spectrum as part of electromagnetic spectrum. Then ultraviolet, X-rays, Röntgen, and so on. So all of them, most of the spectrum is well used, is employed for modern technology. But some of these parts is not fully exploited yet. So this is because it's too high frequency for microwave electronics and too low frequency for photonics, which works in visible and infrared visible. That's why it's called like a terahertz gap. And people from microwave electronics try to push the operation frequency of their components to higher frequency, and the photonic people to push operation frequency to the low frequency to cover this gap. What I am doing, and my team is doing, we are from this side pushing electronic to the top, to the high frequency. So why this is interesting part of the spectrum? Terahertz waves have few very unique features, and some of them are very strongly application-oriented features. And these are summarized here. One is that the terahertz waves penetrate through many materials, like wood, dielectric, in fact. Very similar to X-rays, like Röntgen devices and so on. But the terahertz radiation is not ionizing, so it is compatible with life. And that offers a great opportunity for employing in different areas of human life. Unfortunately, that's only at the beginning of the development. It's not fully exploited yet. This technology just increases its durations. Another particularity or feature of this spectral region is that many materials have particular spectral features in this material, in these ranges. So we call this like a fingerprint. So that means it can be once by investigating or reflected light from these materials, one can conclude what kind of material is that, what is temperature, and what is the pressure. That's very important for radio astronomy, for planetary research, because in order to, as an example, in order to see if there are water vapors in the upper atmosphere of the Mars, we don't have to fly there. It's enough to investigate the reflected light by a radiometer. Okay, and the third application area is enormous bandwidth, which can be used for always increasing demand for data transmission, for internet speed and so on. My presentation for today will focus on these features, because this it's closer to the topic of this conference, like bioengineering, medicine, and so on. So this is also used for space missions, for Earth observation, climate change, which is very popular topic for now, and that's why I thought it could be closer to the topic of this conference. Please, yes. So it worked like this. The sun irradiates the full spectral spectrum, electromagnetic spectrum. It comes to the Earth, it's reflected, but this reflected light, as I told, it has some features on the reflected. And by investigating, if you build such a radiometer, put on satellite cries around the Earth. In such a way, you can investigate what happens on the Earth. For instance, in upper atmosphere layer, formation of cirrus clouds, formation of ozone layer, building the temperature map of water in ocean, and so on and so on. That's a very exciting topic and very important for humanity, because all of us, disregarding of the status, lives on the same planet. And this kind of activity is increasing, and in the last decades became from niche space scientific missions, it moves out towards the technology mainstream and becomes a significant business area, not only in Europe but also around the world. Myself and my team are strongly involved in this activity since 20 years, over 20 years. And all this progress, I know this on my experience. So, starting, let's say, 20 years ago, NASA was the first, ESA starting to develop, and then nobody else was really caring about that. There was not much progress on that. Nowadays, everybody actively working to enter into the space area. China, Japan, India, of course Europe, NASA, and so on. Okay, next slide. The story of this technology is starting in the 80s. So already in that time, radio astronomy, like ESA and NASA, were very interested to get this technology. And in Europe, there are several companies which were building instruments for that, for radiometer instruments. But that high frequency, the only room temperature solid state device which work, it's properly work, is Schottky diode. And at that time, there are no European supplier of this Schottky diode technology. The only supplier worldwide was an American company, it's called VDI. And all the ESA and the European companies were buying these diodes and building instruments on that sensitive elements. But in the 90s, VDI started to build their same own systems. And then ESA and the European company got a problem, so that what if US export restrictions will not allow export of such instrument for European needs? Or VDI will not be able or not willing to export these diode components to Europe. So ESA started to finance, to support development projects in Europe for development of this strategic technology. One of these points for this project was Technical University Darmstadt, where I started. I joined in 2000, in April 2000, starting working on these development projects. And I was one of the first who developed a reproducible process for fabrication of these components. And I'm proud to tell you also my colleague Ivan Lopez, which is sitting there, is joined me for the last 20 years, over 20 years, and actively working in the lab supporting this research and development. But then ESA understood that, okay, now we have this technology, but you are just a PhD at university. So you will defend your PhD and go to industry, and new people who come to university starting everything from the very beginning. So we need a continuous supplying of this. So in this way, I was in the right place at the right time and used this opportunity and founded ACST in 2006 as a spin-off of TU Darmstadt and first European supplier of shortcut technology for telegas applications. Yes. Just shortly, historically, founded in 2006. Then 2013 we moved to dedicated facility for fabrication. Then we push a lot development of system, not only diodes but all the systems. And starting last few years, we are leading European supplier of telegas sources. Next. Next. Okay, so SST, we build components, MMICs. Based on these components, build modules, functional modules like mixers, multipliers, and detectors. And then, based on these modules, build systems like receivers, transmitters, and transceivers. Next. Starting with the diode. The diode, shortcut diode, is a core of our technology. And in fact, we have three basic types of shortcut diodes. One is shortcut diodes which is optimized for capacitance voltage characteristics. These diodes are mostly used for power applications like frequency multipliers where the power is required. Another type is varistors, which are optimized for their forward characteristic, IV characteristics, and with the minimal series resistance and high breakdown, barrier, and so on. That's traditional it's used for mixing application, for heterodyne instruments, for receivers and such, right? And the third type is relatively new, and the ICST, I think, the steel is the only commercial supplier of such kind of diodes. It's called zero bias diodes. We also use the forward characteristic, IV characteristic, but the barrier is very low in such a way that under zero voltage we have still, let's say, measurable differential resistance. For instance, if you have this mixer under zero voltage here, we have the differential resistance in order of gigaohm, which cannot be matched to anything. It's practically it's interruption. In order to drive the normal varistor diode into the operation point, it needs bias. Yes, works perfectly. We can put bias of 0.7 ohm or 0.8 volts. Operate this where the differential resistance becomes in order of few kilo. Yeah, here. And then you can work. But this bias generates current. Current over the barrier generates noise. For instance, a hot electron noise. And that's a drawback for very sensitive detectors, if you want to make a very sensitive detectors. That's why the simple, relatively simple, so in principle, a simple diode, which is called low barrier diode, can solve many problems in terms of receiver sensitivity, linearity, and noise. Next. But this slide is just the same, just to explain a little bit more what this means. So that's typical IV characteristic of the typical silicon diode, PN diode, right? This is typical characteristic of gallium nitride shortcut diode, which high performance gallium nitride shortcut diode. The threshold voltage is at about 4.8 volts, and then we have low barrier shortcut diode, which have a threshold voltage close to 0 volt. And that's the same IB curve which we already discussed in the previous slide. Next. There are other particulars of our shortcut technology, but I think it's too much for today. Due to the time constraint, we will skip this discussion and these two. So now we switch to the receivers. There are three types. In fact, there are two types of receiver technology nowadays, which can be used on satellite. The satellite space missions have particular requirements. So, of course, we have very sensitive superconducting receivers based on hot electron bolometer, for instance, or SIS mixers and so on. But they are usually working at cryogenic temperature. And we just discussed in previous section, yes, it works well, but it has like a system which has to be actively cooling, and it weighs 100 kilos, and to put 100 kilos on satellite is not very simple. Nowadays, satellites are like this. And there are three, therefore people preferred the room temperature receiver, so the sensor which works at room temperature. Shortcut diodes is one of the most sensitive solid state device, which has a mature technology and is working at room temperature. So that's why one type of receiver is based on frequency mixer, based on traditional radio resonant diodes. It has state-of-the-art performance in order of 1000 Kelvin or even below, maybe 500 Kelvin, 300 gigahertz. The problem is that it needs a low chain. So the mixer it means it compares the local oscillator signal with the RF signal and mix it. So that's why we need a strong local oscillator signal, and that's a drawback of this technology. It's not a drawback, but it's difficult because it consumes power. State-of-the-art receiver consumes about 4 watts at 170 gigahertz, like that. If you say, if you discuss about higher frequency, then much more power consumption is to be accounted. We at the CST have developed in recent years a particular new type of mixers which are based on low barrier diodes. And we demonstrated that this technology can use 10 to 20 times less DC power than in comparison with this technology. But it has a little bit more noise. We will discuss in the next slide. And not yet. But, yeah. And the next type of radiometers, receivers, is based on direct detection approach. It is very simple, low cost, and low power consumption. It's about below 1 watt at 300 gigahertz. We will discuss this in the next slide. So that's a typical architecture of a heterodyne receiver. As we discussed, we have the mixer itself. That's the sensor. But in order to bring it into operation regime, we need that the part of the circuit which is most power hungry and very complicated, and also it's difficult for space reliability, space qualification, because the more power we have into the device, the higher its operation temperature, and the more severe are operation conditions and the lower reliability. Okay, good. As I told, we have traditional mixers and low barrier mixers. We conducted a comparative study at ACST. We built a receiver at 300 GHz with traditional and with low barrier mixer, and we observed that the noise of traditional mixer, it's below, it's let's say in range of 300 Kelvin. It was not dedicated. So if you make a space mission, you make a narrow band receiver which is highest performance. Here we just took a full band receiver which is not highest frequency. I told you if you look for highest performance, then we will be at around 1000 Kelvin here. This full band, but for comparison, it's okay. So we have here about 3000 Kelvin, but we need a 10 milliwatts of local isolator power. When we measure the low noise, low barrier mixer, we get twice as much noise, but 20 times lower local isolator power. When we simulated by calculation, the DC power consumption for traditional and for low barrier, we got this plot. It's about 20 times less DC power is required for a low barrier mixer. This includes already the LNA, which has to be included into this. So, and this difference is crucial for satellite space missions because power consumption of satellite, you can imagine, it's a critical point. Next. And that is the direct detection radiometer, which has been developed at SCST for AWS space mission. It is like this. So we have just detector itself here, and LNA in the front. So there is no local oscillator. So it is less complex. Total power consumption below 1 watt. Excellent noise performance because this LNA are already noise performance comparable with heterodyne mixers. The only challenge is that the LNA is not it's limited in frequency. The current LNA technology is limited in frequency. In Europe, there is a technology which reaches 250 gigahertz. At higher frequency, we cannot use this technology anymore. But for this frequency, most of the radiometric channels interesting for radio astronomy are below this frequency. Okay, next. The story of this technology starts in 2009 when ESA had in mind development of one of the biggest space Earth observation missions of ESA was called METOP second generation. It is meteorological operation second generation. It's written like this. And in Europe there was no such technology. And nowhere in the world. That was a pioneering work of ESA to implement, to prepare this technology for space application at such level. So we got the first project on this development on detectors measurement in 2009 in cooperation with Airbus. At that time it was Astrum EADS Astrum. Now it's recalled, it's called Airbus, and now it's again recalled, it's called ADS, Airbus Defence and Space. And other companies, these and so on. So then this, when we finished successfully finished this project, was started another project. We demonstrated very good results, but I think if you consider space application, the technology has to be mature. You cannot send a first prototype of the satellite, because it costs a lot, and if the prototype it has to be mature, maturated. We got another project for maturization of this technology. Finally, we entered into the procurement phase for METOP second generation. It was 1520, and it is not written here, but maybe on another slide. The first satellite with this this radiometer on the board has been launched 1 month ago in end of August. So started 2009, launched in 2025. Okay, 16 years. So just for you to imagine what means high PA space mission. PA it means product assurance or quality assurance. So this means technology has to be maturized and everything used. So that's a kind. I can disclose to you that now ESA is already entering into preparation for method 3rd generation, which is planned nowadays. Is planned for 2035, I think. And we at the CST already got a first project development for development of 1.1 terahertz source for 2.2 terahertz receiver. Next. Yeah, it's just some data for this radiometer. The heart of this radiometer is the detector itself, which has been provided by CST using our diode technology, and that was, yeah, the rest is here means LNA technology is also critical, but this is done by other partners. I can discuss very long about this process and explain you how nice is collaboration with different European companies, partners, and so on. It's very nice collaboration. It's nice to work in this area because everybody is proud to contribute to such missions. Okay, next slide. So this is another slide also for method second generation. So another aspect, it's reliability test for space qualification, for mission qualification, because every mission has own requirements. Operational time, storage time, and so on. Or for instance, for satellite, depending on which orbit it flies, for instance, if it flies around the Earth, it comes either into the sun or into the shadow of the Earth, then this temperature range. So that means it's very hot to very cold, and that this imposes, of course, particular requirements for thermocycling cycles and so on. All this, or vibration shock for rocket start, launch, and so on. All these tests are very carefully prepared. Requirements are prepared, tests are performed, and analysis are very carefully analyzed. Results are analyzed. So that's just some results. That's for vibration shock. This test was prepared for TVAC, temperature vacuum test, for instance, changing the temperature in vacuum conditions, and other measurements, and so on. Just some illustrative examples. Next. Another ESA mission is AWS, it's Arctic Weather Satellite. It's a very interesting mission. It follows the technology platform developed in method second generation, I just saw the previous slides. But the purpose of this mission was to demonstrate a low PA mission. So that means the price of this IWS satellite is maybe just a small portion of the total price of the method second generation satellite. But it was the goal of this AWS mission was to demonstrate a low PA, so low cost satellite, and then, if successful, the following up process is called EPS Sterna, which means a batch of 22 satellites have to be rebuilt in the same way to be launched for weather forecast and planetary research. And this satellite has been launched in summer 24, and EPS Sterna is going to be started by the end of this year, but to last information, it probably will be again shifted, and we will start at the beginning of next year. If you are interested on this technology, you can take this link and read a full article about this mission. Next. Yeah, I already presented this slide. For this AWS ACST, my company with my colleagues which are here, Arthur Negrushan, Joel Opera, and other companies, other colleagues of course, we are very strongly involved, working hard work to develop this and to make all the all the tests, reliability tests for specific qualification and management and so on. Yeah, next, I think, otherwise I'm ready to another some illustrative slides for this mission. There are also other missions, next slide. For instance, HIMS, which is this is not an ESA project. It's a National Oceanic Atmospheric Administration. So we are collaborating also with NASA and other JAXA. JAXA is a Japanese space missions space agency. Then we also have collaborated with Chinese, and so it's a lot of information. I asked organizers of this conference to give me a plenary talk to discuss about all this nice development in Europe, but they told everything is already booked and there is no place for me. Okay, then that's I have to limit myself in this short time. Okay, next. Do you have anything else? Next slide. Yeah. You can, well, next. So, here just the conclusions and outlook, and I want to thank you for your attention.
Just a short question. I think more of a comment. NATO had priorities for detection of plastic-based explosive, and what you have presented, the fingerprint actually matches in gigahertz. So we conducted exercise in Ukraine and also in Algeria. So I'm not sure if you had experience of plastic-based explosive detection using gigahertz.
I remember you talked yesterday, and I think you was talking about this. It's was somebody else, I don't know. And yeah, it's a very exciting topic. But I have to mention. My team and myself, we are focused on technical part, right, and not on scientific part. We cannot cover everything. We are a company, commercial company, not a university.
Is it a continuously variable frequency range, or you focus on specific frequencies?
So the MEDOP second generation has, I think, 12 or 18 frequency channels, starting with few gigahertz and ending with 664 gigahertz. So, and every channel has dedicated instruments, and every partner has the task. We are focused on ICI, on high frequency parts, starting with 89 gigahertz, 180 gigahertz, and up to 640 gigahertz. For lower frequency, there are many companies which have excessive expertise on this area. So we cannot compete on price with companies which can do very good, something at 5 gigahertz. But this company cannot do anything at 200 gigahertz, and that's why our
Thank you. Yes, one question about, thank you, very nice communication, but I have a question regarding the antenna, receiver and sender. Of course, just it puts a problem of cooperative antenna. Super antenna consisted from small antennas, in which the signal are amplified and increases the efficiency of such antenna. Is used, I don't know, I don't observe any communication about such antenna in terahertz region.
I think that's a little bit particular area of technology development, global technology development, and not necessarily a part of this talk. We can discuss about this very nice, but I'm afraid I'm out of time. Thank you very much.
Thank you.
Okay. Then we have the next talk in this session. With the topic of formation of zinc oxide buried layers within the bowls of the aerogallonitride microtubes with Tudor Braniște. Is Tudor here? Tudor Braniște? So, anybody else from outdoors would like to present this talk? Okay, then you were happy.
Everything is on time.
You are lucky, yes, exactly. Now you are on time again. Almost. So the next talk is entitled Performance of Argent 2S zinc oxide nanostructure in radiation monitoring from Maxim Kiriak.
Maxim is busy today, so will be serious.
Okay.
One of the co-authors.
Oh, okay. So, please.
Thank you for your nice introduction. Dear colleagues, dear guests, let me introduce and show some of our current results on the sensors for the radiation, and specifically in this work we are talking about ultraviolet radiation. We know that the zinc oxide and his band gap fits good in ultraviolet range, and even intrinsic zinc oxide acts as an ultraviolet sensor already. Also, it would be nice to have more controlled spectrum and to have an option to maybe selectively set some plus-minus spectrum for the nearest wavelengths. So in this work we are using zinc oxide plus cadmium, which is not set here, but it is explained, and argentum 2S as a functionalized material. Yeah, a couple of words about the importance of this work. We know very good about the ultraviolet benefits and some bad parts. It is widely used in the agriculture, it is widely used in the food technology, in the medicine, and so on. Also, we know very good that it may harm and our health and animal health and the globe in general. So it is very important to control the intensity, the wavelengths, and how it spreads through the atmosphere. That's why it is important to have sensors and biocompatible for the food, for example, some general sensors for the environment monitoring, and so on. And the part of our work is about this sensor. This is the standard checklist, standard program to develop this kind of sensor. The initial zinc oxide we get through the chemical back deposition from the zinc SO4 and the nitrogen OX, which is heated after, with the cadmium, as I said, introduced, that we have the zinc oxide plus cadmium in it. It is thermally heated afterwards, and the second step to install the argentum 2S nanostructures on the top. We create the argentum nanostructures from the argentum SO4 plus I don't remember the exact name of the chemical part. I will answer if it is important. And the resulting nanostructures and the ethylene glycol, we just drop cast on the top, and after we heat the surface, we just get the sensor plus contacts which we deposit on the final step. The structure we get, we have to control its properties, to investigate its properties, its sensitivity to the UV and some other wavelengths. At the moment, our installation of measurements and of investigation, it's on the development, in the active development, so we don't have the wide range of wavelengths to check it, to investigate it. The setup we have at the moment, it's kind of cassette with multiple LEDs, with some wavelengths which we know and which are adjusted for the controlled power, which is controlled with a stepper and the PVM driver, which gives us the opportunity to set the necessary wavelength over the sensor. The another part is heater, which is on the basics of the PL-2 model. Normally this heater may be changed to any other type of heater, like optical heater or some resistive one. Why we choose the PL-2 in this work? Because usually these kind of sensors do not require some high temperatures, and PL-2 gives us the opportunity not just to heat, but to cool the substrate if there is a necessity. And if we set up the correct control, often we need not just to have the opportunity to add some energy to maintain the constant temperature, but also to extract some energy, which we have only with the PL-2 model, with some resistive one or with some optical heaters where there is no such method. Some other things are just for the control, for the communication, which is not strict important in this case. Next one, please. Some results, if we are talking about the sensor and the substrate, with SME we can see the surface, it's quite homogeneous, and if we look at the 200 nanometer scale, we can see some structures with argentum-2s nanostructures on the top. Talking about the homogeneous, we can see with ADX image the distribution of argentum-s nanostructures on the top, and here we have some scan line of the argentum-2s nanostructures and its placement on the top with pretty good adhesion. So the drip cast method to deploy the structure is not ideal, as we say, because it's just standing on the top after the ethylene glycol was evaporated. Also in this case it's good enough, and the sensor shows some stability. We didn't investigate the stability on the long time range. Also in the range of days, months, it's okay. Next, please. This is the response. At the moment, as I said, we don't have some wide range of the wavelength, and normally we will move from the separate LEDs to the monochromatic light source to have the possibility to change it continuously, to control the power, to check not only some specific wavelengths, but the spectrum in general. Also, at the moment, it's kind of this. We have a peak which shows us 317, which is very next to the band gap of the zinc oxide, and was measured some times of sensor response and of the time of the relaxation. In this kind of structures, we have noticed not just for argentum-2s, and also for very wide range of materials, that the time of relaxation is pretty large. We can see 350 seconds, which requires some work in it. It is too much for a sensor, for an optical sensor. and we have a couple of ideas, but I think we will discuss them in our publications. And that's it for this work. A couple of conclusions. We have demonstrated a method to create a sensor from the simple chemical biodeposition solutions, which is pretty cheap for the industry to produce. We have shown some selectivity, which requires to be investigated more continuously for the spectrum as in general. Also for the ultraviolet, we can see the good selectivity. We have noticed the clear peak, which can be used as a sensor response for the specific wavelength. Yeah, we will continue, of course, with this development and to develop the measuring setup as well and the sensors as well. Yeah, I would like to thank thank our colleagues from Kiel University, from the Technical University, from the Agency of Research and Development of Republic of Moldova, which I think will continue to support, to make this project in the future as well. Thank you, and I'm open for questions.
So now the talk is open for discussion. Yeah, please.
I have a small question about efficiency of such a, and maybe comparison with other investigation.
Yeah.
If some words, comparison.
The response, as we can see, is like 400, 415 is a ratio of the photocurrent and the dark current, which is not the best. There are this kind of sensors, but not on the Argentum, which shows some better response. But the issue with this sensor, it is very cheap. We don't use sophisticated technique to produce it. The second thing, it's resistance. The current which flows through this sensor is not too large, it's like microamperes. So we don't need a large power source if we are talking about energy efficiency. The big problem with this kind of sensor is requires some heating. It does not work too good at the room temperatures. But we have some experiments with micro heaters and some other experiments with optical heaters. So, well, it's not the best, but it's normal.
Okay. So if there are no more questions, then let's thank the speaker again. So the next talk is entitled Cooperative Generation of Biomolecular Complexes under UVC Radiation, Application in Dissemination and Diagnostics.
This is common communication together with experimentators and theoretical group from my laboratory, Diana Podolianu, Ion Munteanu, Elena Starodub, and me, of course. We work together in decontamination, but some problems connected with generation of biomolecules under the action of ultraviolet C radiation. It's very interesting for construction a new sensors and a new idea in future development. Please, next slide. This is our communication about this next slide. And this is a principal investigation which was achieved in the past 10 years about such a cooperative phenomena between the radicals in interaction with radiation and especially with ultraviolet C radiation. Ultraviolet C radiation is 250, 200, maybe 300 nanometer in this region, of course, it's short region, and this region of lambda, of course, it's included viruses, bacteria, and fungi also in this range of radiation. And such pathogens may be, in principle, manipulated by radiation. Next slide. This is a construction of tubulin. For example, in our neuron system exists such a protein tubulin. They consist from alpha and beta tubulin, and this alpha and beta tubulin form such a system. And of course, this system are like dipole, plus and minus, and because of this dipole may be manipulated with electromagnetic radiation. And of course, this is important because a lot of malady, neuron malady, it puts a problem how to reconstruct our neuron system. And maybe with radiation, ultraviolet C radiation, it's possible such a manipulation, reconstruct in Alzheimer, for example, situation. If it's possible in pulse radiation, in coherent excitation, because like in one hand destroy the biological material, but in other hand may construct the biological material. For example, P-pulse may excite the system totally, and P-pulse may totally destroy the system, passing it into other states. Next slide. This is a tubulin, and of course, some construction for example, for human, it's a setting such an element, and for bacteria, it's only 3 formed for such a neuron system. And of course, in case of malady, they are destroyed. A lot of people suffer from this destroying of such tubulins, neuron tubulins, and problem of construction and destroy of such a tubulin, it's important in presence of radiation. Next slide. Of course, same situation in DMI, because you have very small, very weak bonds between the strength of such a DNA. In multiplication situation, it's important to manipulate with this strength. Next slide, please. Here it describes it in principle between the adenine, cytosine, adenine, thymine, adenine, cytosine, guanine. Exist, for example, 3 dipole interaction, 2 dipole hydrogen dipole interaction, and tautometric problem. It's also interesting here. Next slide, please. And here, of course, it's a problem with cancer of such manipulate and destroy or reconstruct. Next slide. Now we put a problem of analogy between our systems and of course such a biomolecules. This is a microsphere. Around such a microsphere you have evanescent fields. This is a cavity, and cavity may be biomolecules, and you have excitation. For example, you have 2 radicals, you may excite in Raman excitation such a 2 radical, and you obtain biomolecules from such a two dipole, 1 dipole and 2nd dipole. And of course, if you have such a resonator, so it's possible in such a resonator, such a couple between 2 radicals, 2 subsystems, and create, of course, 3 subradicals molecules. Next slide. This is really, in our cells exists such a spherical for micro resonator. For example, this is a nuclei, and if you look to dielectric constant of nuclei, it's about 1.3, 1.4, and we may have here Galilei modes. The same situation in micro resonator, in photoreceptor, in photosynthesis, for example. We also have some disks, and here is possible the localization of radiation, and this system are similar to our optical system. Next. Our optical system consists from fiber, heavy evanescent field, And the molecular trap in this evaluation field, according to Ashkin, laureate of Nobel Prize, which proposed tweezer effect in which the electrical molecules may be trapped near evaluation fields in spherical situation. If you have a resonance, spherical, microspherical resonator, if you have fiber, and you have propagation of through such a fiber pulses, laser pulses, and such biomolecules may be trapped in these regions and may be manipulated. Next. For construction of dipole, of course it's clear because, for example, you may create dipole in a PN transition, for example. If you excite electrons may be in passing through P, you have formed, but if the material consists from a lot of such a PN transition, you may construct, of course, a periodical dipole system. Next. Of course, in molecules, it's possible such a delocalization. For example, you may excite from these states, electrons are separated like in PN transition in this region. You may create a dipole. Here you may create a dipole. And such a situation is possible between the molecular subsystem, for example, amino acid. Next slide. Next slide. And of course, if you look through all these biomolecules, like that, it consists from dipole. It's like our building. It consists from blocks. Very interesting was in the glue of these blocks. If glue is not so, the interaction between the blocks is very small, it's better to destroy the glue in order to destroy the block. And such a situation is put, for example, in destroying the virus and bacteria. If virus and bacteria consist from DNA and consist from protein, and protein consists from alpha and beta subsystem, it's better to destroy these blocks, and you may destroy virus. Not studying what's happening to the blocks, because we are not interested in many cases what's happening in the blocks, because blocks is a chemical amino acid. It's like optics, for example, for atoms. People which study optics of atoms and molecules are not interested in what's happening in the nuclei. Yeah? It's open what's happening in the electron subsystem. Of course, the nuclei play an important role. So it's clear. Such a situation was puts a problem about packing such a system. For example, you have such a protein. It's possible to represent it through such a 3D poly. You have such a protein. You may represent it from in plane with such a dipole in vertical situation. The interaction Hamiltonian, of course, is dipole-dipole interaction. Next, sum of dipole. Maybe one dipole consists from many dipole. You take macroscopic dipole, yeah? Next slide, please. And you put such a problem of construction, for example, tetramer in plane, tetramer in space. Next. And you put the problem how this excited state may be stable regarding the ground state. Because if you excite the system passes into ground state and radiate. If you excite in the system, the lifetime is very, very high, you have stable situation. It puts a problem in two quanta excitation that these states are dipole forbidden. Dipole forbidden regarding ground state. You have a stable situation. In opposite situation, if you excite in such, you may destroy this molecular system. Here, it's complicated situation. It was developed such a theory. Next slide, please. Here you have tetramer. Tetramer also, it's in one excitation in tetramer, two excitation in tetramer. Stability of such a excitation regarding ground state. Yeah, next slide. Next slide. next slide, because this technology was developed. Next. And possibility to generate such in Raman situation. Next slide. Next, pentamer. Next slide. And this is our pulse technique, connected with our laser, ultraviolet C, and how such a pulses interact with such a system. It's oscillator and of course induced. This is Raman stocks. Is absorbed is the money anti-stocks and so on, or opposite situation. The frequency between the stocks and pump, it's about terahertz, because in terahertz region, Professor not emphasized, but in terahertz region is very complicated to have a laser. And in Raman situation, it's possible difference between hertz, between stocks and anti-stocks to obtain the frequency in terahertz, which are in resonance with such a weak coupling between the dipole. And this is very important in the Raman situation. Many people don't know about this. Next. This is our equipment. It's connected with such metamaterials. And here it's constant with ultraviolet C, 6 lamp, and this is here is metamaterials penetrated by gas, contaminated fluid, and so on. And this is our equipment. Next. And this is our measurement, 1 lamp, 2 lamp, as function of intensity. We introduced 1 lamp, 2 lamps, 3 lamps, 5 lamps, and construct the dependence of the contamination as function of intensity. Next slide. And this is metamaterials like balls. Of course, here it's about 1 millimeter, because the fluid must penetrate. If you take very small balls, the resistance of fluid is increased. But of course, this is possible, and we introduced fluid from one part to other part of this cavity. This is a penetration of radiation from one pulse to other pulse. Increase the contact surface. If your fluid are not transparent, if you radiate with ultraviolet, you have depth of penetration. Depth of penetration is very small. The fluid may undecontaminate. If you introduced in this fluid our pulse, of course, they communicate with radiation from one to other, like in periodical optical structure, and they transmit from one pulse to other pulse to other pulse. Next slide. This is our pulse and our equipment. Next. And this is because you pass from continuous to pulse radiation. Of course, we study this situation in interaction with short pulse. This is our NATO ultraviolet C machine, which generate nanopulses in 250 nanometer. And this is a cuvette with metamaterials. Next slide. And this is our dependence obtained by our experimentators. As function of intensity, they have some peculiarity connected with critical intensity in decontamination. Next slide. Of course, this pulse consists from set of pulses, because resonator, some pulse consists, if you will do the Fourier transformation of this pulse, it consists from other pulses separated about one to other about terahertz frequency. And of course, stocks and anti-stocks exist in such a pulses. It's possible this to demonstrate. Next. This is our machine, and this is Fourier transformation of our pulses. This distance is about C divided by 2L. L is a distance about maybe 5 centimeter. This cavity of pulse radiation. Next, and thank you. Next. This maybe is here You propose another effect, not Ashkin effect, connected when it was this pandemic. We continue to work in this direction, and you proposed to measure the decontamination efficiency as function of velocity of fluid. You propose another effect connected with the rotation of fluid between such a balls. It's like radar, mountain radar, which flow from, if they flow very, very high, of course, the decontamination and the fluid become very, very good. And if you have here a particle, and the density of particle is higher than the density of fluid, you have like centrifugal. The density particle will leave this smerging. In this situation, it's possible to have a contact between the nanoparticle and surface of such a balls, which are penetrated by ultraviolet radiation. Thank you. This effect was it's all. This equipment, of course, and thank you for your conclusion, and thank you for your attention.
Let's
Yes. It'll take only 1 second. Are you allowed to handle prion in your laboratory? You mentioned prion. I'm not sure if you're allowed to handle prion. Molecule in your laboratory.
What's it?
We'll talk later. You have mentioned prion. It's almost impossible that you will be allowed to use prion in your laboratory. So that's the reason I asked.
Maybe it was like a short question, but
it's a biological molecule. Unless you have a biosecurity laboratory, you cannot handle it. That's the point I want to say.
Thank you.
Professor Yanaki, I want to thank you very much for a very exciting talk, very interesting. And just a comment from my side. You told that there are no lasers or terahertz frequency. I want to say that we at ACST produce these coherent sources with accuracy down to 1 hertz. So that, if you need some equipment and terahertz, just contact us.
I know it's very important.
Unfortunately, the time is running, is pressing. Let us thank the speaker again. And try to look if the next speaker is available. So the next talk in the program is written comparison of emission properties of water, soluble mono and tetra P-cent derivatives. So your name is Tamara. Okay. Thank you very much. Please proceed.
Thank you for your presentation. The title of my presentation is Properties of Water Soluble Mono and Tetra Carboxybenzamidomethylen Zinc PCA Derivatives. Metal phthalocyanine find widespread application in as paints, colorants, and other, but presently this phthalocyanine are used in the photodynamic therapy as photosensitizer for treatment the cancer. Next. My presentation consists from a method of synthesis, structural confirmation, absorption, emission, and transient absorption investigation. Next. It's well known that cancer is fatal disease being the second most common cause of death among people. Globally, the number of cancer cases is expected to reach more 28 million by 2040. Presently, there are a lot of
cancer treatment methods such as chemotherapy, radiation therapy, immunotherapy, hormone therapy, and photodynamic therapy. And sometimes is used for treatment combination of this method. I will continue with photodynamic therapy because it's a treatment that uses light-activated drugs called photosensitizer, a source of light and molecular oxygen that induce diffuse cancer cell. And here is illustrated how can be destroyed cancer cell. Current research focuses on the second and third generation of photosensitizer, which include various phthalocyanine derivatives. Phthalocyanine are well established in photodynamic therapy because of their strong absorbance, higher singlet oxygen generation, and photostability. Next, please. The main characteristic that possesses and photosensitizer include low duct toxicity, high phototoxicity, high selectivity, rapid elimination, absorption in the critical window of tissues, higher singlet oxygen quantum, water solubility, and physiological stability. Next. In this presentation, I will discuss more about zinc phthalocyanine because our well-established photosensitizer and photodynamic therapy due to their strong absorption in the red region, photostability and high efficiency of singlet oxygen generation. One of these advantages of all metal phthalocyanines and of zinc PC is bad solubility in water or organic solvent. Therefore, for improved solubility, we functionalize it or substitute it with different growths that are not toxic for human body and used for further investigation for uses in photodynamic therapy. In this presentation, we use a group that is carboxybenzamidomethylene group, and we compare in this study photophysical properties of the zinc PC derivative with single group and with 4 group. Next, please. Why we choose this group for substitution? We use because enhanced solubility of the metaphthalocyanine, extinct P-conjunction with other odds shift in the red absorption in the region of tissue. Improved quantum yield of fluorescence and reduced in persistent processing, reduced aggregation, and after substitution with this group, we can conjugate with different materials such as polymer, nanoparticles, or other biomolecules for targeted therapy. Next, please. And here is a scheme for the synthesis of mono zinc PC derivative and tetra with carboxybenza-midomethylene zinc PC. For the synthesis was used phthalic anhydride and metallic salt, in this case zinc salts, and synthesis consists from few steps. Next, please. Activation, addition of paraformaldehyde, addition of phthalimide, and cleaning. Next, please. And for the characterization, we use absorbance, fluorescence, and phosphorescence spectroscopy. Next, please. Here is presented absorbance of zinc PC derivative with single group of carboxymethylene and with tetra, a full group. And we can see difference between position of the absorption band. The addition of more group in the zinc PC derivative shift the bands in the red region. Also extend P conjugation and such improve better tissue penetration in the penetration for photodynamic application. Next, please. This compounds emit, and here I presented fluorescence at excitation with 300 nanometer, and one can see for mono, for both mono and tetra, one broad band with some featuring and less intensity band in the red region. To understand this band, we used for the excitation another wavelength of 640 nanometers, and we can see that our compounds emit in far infrared and with emission in far red region. That is good for application in photodynamic therapy and for better penetration in tissue. Next, please. And here measured room temperature phosphorescence fluorescence lifetime of both compounds, and we obtained the lifetime of the excited states for tetra, average theta about 5 nanoseconds, and for mono 3 nanoseconds. And we can conclude that with increasing the number of substituents group, increase fluorescence, increase the lifetime of the excited state, increase the quantum yield of the fluorescence from 10 to 22%. Next, please.
2 minutes.
And also our compounds emit phosphorescence in the all visible red region with a broad band and of higher intensity, and two with more or less intensity. We can conclude from this investigations that in the case of tetra substitution, we have more efficient triplet site formation in both compounds. And this allows us to use it in photodynamic therapy and bioimaging. Next, please. And transient absorption spectra confirm that more triplet states are present in the compounds with tetra growth. And this feature indicates that triplet state population is relevant for photophysical application, including photodynamic therapy. And the comparison of mono and tetra substitution highlights that the more substitution with more group improve photophysical parameter such as fluorescence and lifetime of the excited states. Next, please. And conclusion. Both mono and tetra zinc PC derivatives show significant absorption in the tissue transparency window. 600-400 nanometer that associated for photodynamic application. Fluorescence yield increase with increasing number of the substitution growth from 10% to 22%. And both mono substituted and tetra substituted exhibit phosphorescence. But transient absorption spectroscopy confirms that the more efficient excited state dynamic is compound with tetra substitution. Because present triplet, more stronger triplet state formation. And thank you for your attention.
So I have another question. You told us about frequency window in the tissue
transparency.
Yeah.
Our body.
Between 600 and 800 nanometers. It is basically red color. Can you comment a little bit how deep we can penetrate less?
10 micrometers.
10 micrometers.
Yes.
10 micrometers, not millimeters.
Not millimeters.
10 micrometers. Okay.
10 micrometers.
Okay, it's not very much indeed. Okay, but in fact,
we need more
penetration. You can think about conducting this layer light by some insight, like
yes. With intravenous.
Very interesting talk. So, any questions to speaker? Okay, in this case, let's thank speaker again. Good. Well, personnel, we still have, according to our program book, two more presentations, and the next presentation is effect of PTFE thickness on gas sensing properties of titanium dioxide palladium doped zinc oxide nanostructures, presented by Professor Lupan.
So I hope the conference dinner will not start without us, so you can stay. You can stay a little more longer here. And since everybody is tired already at the end of the day, but let us try to focus on the last two presentations. I know Professor Lupan will give a very dynamic presentation. Let's pay attention.
Thank you. So, dear colleagues, so I want to show some results of our PhD student, Mihai Brânză, with all co-authors, about covering sensors, again with polymers. In this case, it's PTFE. So it was one time when PTFE was tried to get rid of it, but now it came again in focus of applied science and applications. So next, this is joint work between Department of Microelectronics and Biomedical Engineering, between Department of Material Science, Chair for Multi-Component Materials from Kiel University, Functional Nanomaterials, Professor Adelung was here, and PCL University from France. So we know, explained already, but everybody wants smart houses, smart farming, smart city, everything smart, a lot of industry, good health care, and so on. But everywhere are required sensors. They should be precise, cost efficient, multi-purpose, and especially for biomedical applications, it's important to have very high sensitivity, high selective, and to be used on different environments. And also to not be affected by water. So in this case, PTFE is a very good one. As we mentioned, the goal of our, one of goal is detection of different types of exhaled gases from PREF. So this is a very good marker, biomarker, to identify diseases in very early stages or even worse. But it's a non-invasive technique, which is highly promising to start to develop new types of devices and even for longer distance identifications. For this, next we go to our proposed technology for these devices. So we used zinc oxide doped with palladium. We already reported previously, it's a very good sensor. This one, stable. And it was made by our technology. We possess all of this. It's made from different precursors of zinc sulfate, hydroxide, on different types of substrates. It can be glass, it can be ceramic substrates. Doping of palladium was very simple from palladium chloride in complex solutions on top to protect the surface from other environmental factors. It was deposited at ultra-thin TiO2, which is highly stable material, about 10 nanometer thickness by atomic layer deposition, and another deposition was made by spray pyrolysis to create some nanocrystals. On top, nanograins, which is highly important for sensing applications, and afterwards it was deposited gold contacts and used our technique here to investigate sensors. So PTFE, we have here identifications and how looks the structure of this molecule. So we have carbon atoms and covered from fluorine, as mentioned. It's trying to get rid of this, but now again people are searching for this technique to find them and to use in various applications, including biomedical ones.
which technique to be deposited. So if we want to make some sensors on substrates which cannot be heated, for example, or flexible ones, it's very important to have a technique, like I mentioned, a new one, ICVD, which can deposit at room temperature. High quality PTFE layers, pinhole, free, so heating filament is only 60 watts. Adding polymers is deposited this type of polymer on surface. And since nobody studied these structures, we investigated firstly this current voltage characteristics. It was initially studied at different operating temperatures, but after some times we saw some changes in these characteristics, which means we are still not so stable at high temperatures. But for sensors of these materials, and it was one question, depth of this penetration, another presentation about these polymers. It's also still investigating to see thickness of these layers. Usually in previous studies we used films of polymers and copolymers about 20-25 nanometers. So at this thickness we still have a good detection. In this case we investigated for PTFE samples with 40 nanometer on top, and we found that hydrogen still can pass through this layer, and it was detected at 250 at high temperature. Since was observed that it starts some changes in this polymer structure, then we can see that all different types of gases and volatile organic compounds can be detected. After 3 months, also it can be seen, but sensors are still alive, they are working. 250 still show high selectivity, only hydrogen detection, and at higher operating temperatures structure is changed, but still sensor detection. For a thicker layer of top PTFE, like we wanted to see this penetration. So we cannot detect so well compared to our under 250 no detection of hydrogen, so it looks it cannot penetrate this polymer. But after 3 months and after a lot of measurements, already it looks it's creating some pinholes through, some holes through polymer surface, and hydrogen is detected at 300 degrees. Some other types of or combinations of this material, why PTFE? Because it's very good and hydrophobic, so doesn't allow. In this case we use these hybrid materials by combining PB3D3 with PTFE polymers, so it's called copolymer in this case, and it was studied at different relative humidities. So if for example clean room or some other environments where it's really low humidity, about 10%, we have detection of hydrogen for this mixture or copolymer. For hydrogen at 300, at 350, and for other gases it's really low. But at higher humidity environment, or about 45%, in this case only selectivity to hydrogen was detected. As I mentioned, for all of these types of sensors, we are publishing one by one in scientific papers results, and done all these calculations. And it was observed, for example, ring structure for initial molecules, but after annealing, for example, it's formed cage already structure of these types of materials. And of course, these types, they are showing different selectivity. So in this case also at higher temperature, but starting from 250 degrees operating temperature, we have only hydrogen detection. And for cage, this is for ring structure. For cage, we have really much lower response for this type of sensors. Another ones are showing only, for example, with PB4D4 polymer, in this case, and also it's forming these types of rings. And in conclusion, these types of devices can be created, can be used or planned to be used in various medical applications and also in space and other types of devices to protect from environment. So this is really a broad application. Next, thank you very much to our team in Moldova, of course to Professor Shonte, main organizer of this conference and head of this conference. We wish him a good health. And to our PhD students, already some of them are PhD or doctor in science, and of course of our team, a collaborative team of Professor Adelung. You already know, like I mentioned, a single professor and 40 PhD students. It's like a big institute because we're the largest group and, at this university, and highly active, growing, still growing. And acknowledgement to Professor Falkel, which is focused on these types of polymer growing and trying to implement. We already made a startup company with these types of setup, so it's planned to make a selling of this type of new device for ICVD.
Thank you very much. Yes. Professor Pan, you told in your presentation, it was a very nice, very dynamic presentation, as expected, in fact, right? You mentioned that the optimal layer, the thickness of layer for your ICVD polymer is about 25 nanometers. It is really, for polymer, is not very much, right? Can you comment a little bit on the surface roughness, variation, thickness variation across the wafer, and reproducibility of this thickness?
So it's highly reproducible, and it can cover any types of substrate. I will show on plenary talk how it can be covered with nanometer range, any shapes, and it's covering everything. So no holes.
Okay, and so the surface roughness
at this stage is not so relevant. So technique is really, because it's something like a gas phase.
Okay.
Deposition.
So this you mean that the surface is fully covered already by few nanometers of thickness?
Yeah.
Okay.
But we started to use 25 just to not get affected by some other volatiles, compounds, some other gases, to not penetrate, to not interact, and to change selectivity.
Thank you very much. Other questions, please?
Professor Rupan, it's a very interesting communication, but when I work in Optoelectronic Institute of our Academy of Science, we meet such a situation, competition of sensors and so on. Optical sensor, if you want to measure some gases, some property of maybe, I don't know, not so good cases, exists such an optical sensor in line of absorption and so on. What is correlation? Your sensor and optical sensor. You know
the price.
What is efficiency, correlation in the literature, of course, in literature, of course.
So firstly, So
because if you want to put your sensor in production, you have your costs.
We try to develop all these complete technological steps, so we have complete line of technology for production just in case, and trying to have all this as much as we can do here in Moldova, but also in Germany with our colleagues, some parts. And in all our reports, like I mentioned, we are trying to focus mainly on new types of structures, hybrid structure. So that's why, and always we are comparing with literature. Now, every reviewer and scientific papers during review, we are asking, show comparison with other. So we are showing 2 pages tables with data and mention. This is our parameter, which is much better than previously reported ones.
Maybe.
Yeah. So always we are trying to highlight this.
But also I think this is just a piece of maybe quarter of millimeter small sensor, and it costs maybe half a cent,
maybe,
right? And an optical sensor may cost
several hundred.
Sometimes
Optical so much, but also we are trying to study this optical properties for our sensors.
Yeah.
Like our students showed some
transparency and
results.
so on. Yeah.
It's very important because also by coverage we can this type of coverage we can get rid of ambient factors. So optical sensors can work really very good, but we still searching, trying to involve young generations to do this.
Okay, thank you, President Manuel. You nice talk. Let's and now we come to the last talk on this session on this day-to-day impact. Is entitled 2 qubits in thermostat. 2 qubits in thermostat by Artur Rotar. Okay, thank you. Yes.
I'm Artur Rotar. I'm a master student at Moldova State University, and I also work as a junior researcher at the Institute of Applied Physics. Today I'm going to be talking about the system of 2 qubits in thermostat. And before starting, I want to mention that this work was only possible because of the supervision of Dr. Mihai Makovei. And before I start, I want first to explain what the word qubit means and what the word thermostat means. So qubits, basically, it can be any quantum system that has 2 levels. For example, in my talk, I'm going to be talking about a 2-level atom that can either be excited or it can be in the ground state. For another qubit might mean, for example, the spin of the electron, which can be either up or down, yes? Or might be, for example, the ionization of an atom. Either it's ionized, either it's not ionized. A thermostat, another equivalent term for thermostat is heat bath. Heat bath is basically a very large system that is only made from electromagnetic radiation. Like, for example, we're in a heat bath right now because we have electromagnetic from the lamps, from the projector, from outside. Next. Why this work would be important? Is because where I'm going to mention a little bit later, we're working in the open quantum systems. Open quantum systems, basically, they're not isolated, but we have interferences from the outside, which is like the thermostat in the talk I'm going to give it now. And why this is useful, not just for theoretical talks, but also in practice, because as from other articles here cited, like contrary to common sense, usually when we want to make the efficiency of a motor, of a heat engine, or of a refrigerator as big as possible, we think that we need to isolate it as much as we can from the noise coming from the outside. However, the article is showing how we can have higher efficiencies if we take the correct steps to integrate our system with the environment. Higher efficiencies, or we can have, for example, entanglement that persists even though it is decoherence in the system. Next. So here are 2 things I want to start. The first of all, as I mentioned, we're working in open quantum systems. The system we want to investigate in this, we want to calculate the population of our, like what is the probability to get the ground state of the atoms and the excited state of the atoms. And the environment is the heat bath. And here, the first step that we need to do to be able to analyze the system is to write the total Hamiltonian of it. And here, this is the total put in one place, and I decompose it so it's easier to see. The electromagnetic field, which is like AK and AK dagger. AK means that photon is destroyed, or operates for the destruction of a photon. AK dagger means the creation of a photon. When they're put together, it means just the average number of photons that we have in our heat bath. And the H omega k, of course, means like the frequency of our photon, the amount of energy. Why is here a sum over k? Because in this environment we have an infinite amount of degrees of freedom. Because I can have an infinite amount of energies for my photons. I can have more polarizations and also different types for the photons. Now this is the atom part of H omega SZ. SZ is basically the difference between the levels, yeah? Because, for example, if my ground state is for 2 electron volts and my excited state is 10 electron volts, I'm only interested in the difference between them. I'm not interested in the actual absolute values that they have. And this is the most important part of the equation, which S minus basically means that we have like the from excite to ground, and S plus from ground to excite, yeah? And this is basically, when I'm having my 2 atoms, for me to know if they're excited, ground excited, which configuration they can have, I need to measure, and that's going to influence my system, and that's not what we want to do. And because of this basically term is, like, for example, the first atom goes down, the second one goes up. And I still have the same configuration of one excited and one ground, but they just switch places. That's the index. This is written for n atoms, but in my case it's going to be just 2. And this is just the interaction term. This is the easiest one actually to explain. S minus means the atom goes from the excited to the ground state, and of course, you know, we have to have energy conservation. It means that then we have to create a photon. Yeah, with the photon, it's going to have the exact the same energy as the difference between the levels. And here is the other way around. If a photon is getting destroyed from my system, it means it was absorbed by one of my atoms, which means it has to go from ground to excited. Actually, there could be some more terms here, but we're neglecting them to have conservation of energy in our system. Next. Okay, so here is the my two-level atoms. And after we have written the Hamiltonian, we want to, at the second step to solve our problem, we want to change the basis. We want to work from the simple basis of excite ground. We will have one of them simple, when both my atoms are excited. We'll have them when both my atoms are in ground. And then we'll have a symmetric and anti-symmetric eigenvector of the system. This one is symmetric because of the plus, and you see it's like the first one excited, the second one ground, the second one excited, the first one ground. But I don't really care which one is which, because the atoms are identical. That's why they're put in the same place. And here I have a minus because of the minus sign, I have the anti-symmetric one. Yes? And after I am using this new basis, my operators of the first atom getting excited, the second one atom getting excited, I have to rewrite them. Which is basically the same thing physically, just a different mathematical way to express the same things. And this is how my Hamiltonian from the previous part. Could you please go back? So we've turned this, again, to this, by using this mathematical tool of changing the basis. So how does this look? Not in formulas, but in, we've changed from two ground 1, ground 2, excite 1, excite 2, into, well, like, we've changed two atoms with two levels, like one atom with 4 levels. And in the case that we have studied, we will neglect the antisymmetric level. We can talk about that later, so just to save time, you can ask me if you're really interested. Next, why it's neglected. So here is like the gist of the open quantum part of it. Why is it, I'll explain it fastly. So we can, because our systems of atoms are coupled to the environment, we cannot have pure Schrödinger type equation, because in the Schrödinger equation we must have a pure wave. But here we cannot have a pure wave because our systems are coupled to the thermostat. This is the equivalent to the Schrödinger equation, just when we are using a density matrix and not the equations. And if we plug this back in, we're basically getting this. This is the same thing as this, just rewritten. And the crucial thing from all of this I want you to pay attention is just trace F. What does that mean? Is basically we are going to get, this is a fancy way of saying that we're going to average all the values from our thermostat when we are taking the system we are investigating, yes? So we have this complex looking Hamiltonian equation, but it's like simply said, the interaction between the environment and the system, we're going to average the interaction from the environment and see how our system behaves in that case. This is not that important. Where is the thermal field coming in? At the average value that I'm picking. If, for example, I wanted to work not with thermal light, but with squeezed light, then here will be some different equations. Of course, the angle brackets mean average values for the operators that I'm using. And if I'm, could you please go on back? Get this, plug it in here, we get this one after the integration and after taking this, as you can see. And here are also like the constants that are in front everywhere here. Yeah? And after solving this equation, next please. After, we're going to obtain, because I have neglected the antisymmetric level, excited, symmetric, and ground states. They're actually coupled, a coupled system of ODEs, of ordinary differential equations. But we decided to solve just the case for the steady state, not to get too complicated. And here mu is basically the Boltzmann distribution for temperature. So I'm saying that my thermal field outside my atoms is just the simplest case possible Boltzmann distribution. And why does this graph make perfect sense, that it should be this way? If my temperature is 0, then the probability of finding my atoms in the ground state should be 100%. There's no reason why they should be excited, because the temperature is 0. And the symmetric also 0. The symmetric and the excited ones are 0, yes? Of course, then as we increase the temperature, we see that the ground state begins to drop, and the symmetric state and the excited states begin to become more probable for us to find our atoms in there. And It's not really clear because it's not the same size here, but they all converge on 0.33. So for infinite temperature, I have equal probability in all levels, which makes sense because if my environment acts with infinite temperature, it means like there's no dynamics. I just have everything equally probable. And the last thing that we did, we also calculated the fluorescence spectrum in far field, basically in distances that are far bigger than the wavelength of our thermostat. This is the formula for the spectrum. We've calculated it, and the difference between the normal spectrum and the spectrum where we use the dipole in the Hamiltonian earlier, where I showed that we begin to see the splits. Basically, you see here a minus and a plus delta. Otherwise, everything looks pretty much exactly the same. How would you see this on a graph? Besides your big spike in the middle, you also have two spikes, one to the left and one to the right. And that's kind of all. No, thanks. And conclusions, what we did here. How we did it, we calculated the population and some information.
So, Mr. Rotar, I was a little bit confused from your statement that in order to keep the law of energy conservation in your system, you have to neglect some terms of your equation. I'm not sure what you mean with that.
But okay, that's explain.
That's all right. Maybe I just, it's not so important. I don't want to enter into deep theoretical debates. So please, the question is open to discussion.
I want to put one question, which I obtained such a question from other professors. One professor from French asked me, if you have two qubits, in uncertainty principle, delta n multiplied to delta phi more than 1. If you know delta n, 2, delta n is equal, fluctuation of delta n is equal to 0. Delta phi is infinite. How you prepare two qubits? Is two qubits classical qubits or quantum qubits?
No, they're quantum qubits. Just as you have seen,
because you already declare.
Yeah.
This is a declaration. If I have two particles.
Yes.
If I have two particles, you have undistinguished in the phase.
Yes.
It's clear?
Yes, yes. Yeah, I understand what you mean. We know it's quantum. It's as you have seen, I have not talked about the phase at all in this, because the phase is like infinite because of the uncertainty in the phase is infinite because of the uncertainty principle. So I know that there are two particles exactly, but I have no idea what their initial phases are before the experiment. And as far as I know, they don't matter for this calculation. For the population and for the spectrum, there might be some cases where the phases do matter, but I have not studied them.
I also answered him in such a way, but he asked me to be more prepared in this field. Yeah, I think that you think about this. If you have two particles, if you have one particle, you nothing have about phase. If you have two particles, you nothing have about phase, according to quantum mechanical principle.
Okay. So, any other questions to our speaker, to our last speaker today? Good. Then I think this discussion can be continued, and we still have 1 hour before the dinner. So let's speak the and. That was our last talk. Thank you very much for your great attention and great commitment to stay here until the very end of this session. Now I wish you a beautiful stay and a good dinner.